Siamo in diretta? Quando ci danno il via? Allora, io dico le parole iniziali, poi tu presenti Antonio Muraro. Quando la fai la commemorazione? Mentre parli o prima? Mentre arrivi a parlare?
Okay. Cominci la presentazione.
Ok, quindi noi poi chiamiamo. Possiamo prendere posto?
Please take seat. Good morning, everybody. I would like to welcome also all the participants that are connected online. I would like to welcome you on this conference. We have decided to take into account some important issues related to multiple sclerosis, and we would like to dwell on research on issues that can become a treatment in order to improve the quality of life. Today we're going to speak in Italian, so by doing this, the streaming can be followed by an awful lot of people. I would like to welcome Professor Mancardi, who will be the coordinator of this session, and also I would like to welcome you all in this conference, and I would like to welcome all the speakers that will deal with important issue. The morning session is devoted to stem cells and health brain, and so this session, this conference will be an opportunity to share the last advancements on the three main pillars in which we have invested during the last 25 years. So we have invested funds allocated to scientific research. I would like to hand over to Professor Bancardi to introduce the first speaker. So I think that we can get started with the morning session. So we have the first discussant, Professor Antonio Celli, who worked with me, and he is a professor of neurology, and also he is the scientific director of Policlinico San Martino. He has coordinated the study about the mesenchymal cells, and actually he will draw the lessons of this research. Thank you, Professor Baccardi. Thank you, Professor Battaglia. Needless to say that it's an honor to be here with you. And as a disclosure, if I if I have achieved these results in my career, this is also thanks to the Italian Association of Multiple Sclerosis, who believed in me, who believed in what I wanted to do. So for me, it's a pleasure being with you today, because you are an integral part of my life, a key component of my life. So I would like to say something about a story, a beautiful story that ended in a not so good way. And maybe because actually, so I will present some figures which are really complicated. I will try to simplify those data. Mesenchymal stem cells, stromal mesenchymal stem cells have been exist since a long period of time, and they are considered as cells that can treat the multiple sclerosis and other neurological issues. Why are we interested in those cells? Well, actually, I was intrigued by two works. Here you have a work that has been published, is really sexy, because the title is Turning Blood into Brain. So this is really this is something that this is something, it looks like commercial things, but by the way, this work that has been published in Society, and it has been really criticized and amended partially from a scientific point of view, because actually blood cells was cells of the blood in the bone marrow. They can acquire phenotype, but by the way, this work was really, I was really impressed by this work, because actually I found it really useful. And also there is another work. I was attending the World Congress of Immunology in Stockholm, and I was near Andrea Bacigalupo, who, together with Professor Marmon, he's the father of the technique of the hematopoietic stem cells, and Carlos Stella was speaking, and he told me, I have the bone marrow cells that suppress the lymphocyte proliferation. And so I was reading that those cells can be transformed into neurons. And I said, well, on the other on the other hand, we have we can have cells suppressing T lymphocyte. And they become neurons. So this is fantastic. So that's why I tabled the project to the Italian Association of Multiple Sclerosis, and so this project has been adopted, and we got started. So I will be really fast, because actually I don't have so much time, and as I was telling you before, this is a work of Bruno Bonetti, that we know really well, and he showed that actually they can acquire phenotype, a neuronal phenotype, but actually what they do is to ensure neuroprotection against the neurons that die. So there is something really interesting that is represented, something interesting, because actually at the beginning we thought about the mesenchymal that may induce actions carried out by other elements. Here there is a work of a work that mesenchymal stem cells promote neurogenesis, and this has been confirmed in vivo by has been confirmed in vivo because actually mesenchymal cells were induced in the brain of rats, and actually they saw that those mesenchymal cells did not repair the problem but activated, they were able to promote neurogenesis. So, by the way, my background is a neurological one, so during time, so we have shown actually that the mesenchymal can inhibit the T cell proliferation, and this is due to the fact that when you have inflammation, the mesenchymal cells become more important from an immunosuppression point of view, so they stop their capacity, so they lose the presence of stem cells inside them. The mesenchymal cells have shown that they inhibit the T cell, they inhibit also B cell, and also they block the capacity of the dendritic cell to present the antigens. So the dendritic cell can tells the T lymphocyte what they have to do, and the mesenchymal cell in vitro and vivo in a model, in an experimental model, they can block the dendritic capacity to become lymph nodes and to do the actually this work is pretty old, but the principle is clear cut because actually they told us that at least in the experimental model in vivo, in vitro, mesenchymal cells can block the innate immunity. So here we have a dendritic cell that may be infectious in presenting the antigens, and then they can block the cells of the acquired immunity. So all these things allowed us for the first time in 2005 to show that by injection the mesenchymal stem cells, so we were able to improve the situation of multiple sclerosis. And actually, there is something new because actually that was the first time. But as I always say, when we work on rat, we know that we can improve the development of the disease if we have the right bacteria in the saliva, for example. But by the way, there was so the most important thing is that for the first time we were able to show that stem cells were able to induce in vivo a tolerance, an immunological tolerance, because actually with the lymph nodes of our rats, of our mice, so we took lymphocytes and we used myelinic antigens, and so with the treatment of with mesenchymal cells gave a good result. Actually, we raised the same question. These cells go to the brain. Unfortunately, a few of the cells go to the brain, others go to the lungs. And so another question, if they co-localize with the natural antigens, and the majority of these cells do not do relocalization with those cells, and this confirmed what we thought, because transdifference is really limited. But what we have shown is that nevertheless they do neuroprotection. If we assess the number of neurons that we can find in the bone marrow of the mice, we can see that there are more in animals that are treated with mesenchymal cells. And we said, if we take out into the brain, which is an issue which has been tackled by Gianvito and other colleagues, Dimitri Carusis, if we take out in the brain through lumbar puncture that we can do in the mice, so the concept is the same, but maybe we can achieve better results but the mice is treated through intrathecal method are going better. And so there are no changes from a clinical or pathological point of view. So the answer stems from this work of Bob Miller, Cleveland, that he did something simple and innovative at the same time, because he said maybe there is a secretion of stem cells. So he took the
Scusate, un genovesismo che mi è stato chiesto.
breeding ground. So in the breeding ground in which our cells are, and we inject them into the mice, and we can have the same result as we do the injection of the mesenchymal stem cells. So the most important thing are the substances that we can find in the breeding ground. So these substances are different. This is an old study that we did some time ago with chemokine, which is really important because chemokine is allows the interaction between neurons. This is something important. This is a neuroprotection factor, and the secretion is from the mesenchymal cells. And then, for example, in this case, there is a study made with Claudia Varderio, and we showed that administration vesicles produced by mesenchymal cells can act in the microvia. And so this is what I was saying before, because actually mesenchymal cells give the instruction to other agents to do their job. So microvia, for example. So this is a recent work. Tiziana Vigo in the laboratory has shown that the mesenchymal cells make astrocytes good. This is something that can be done in vitro, in vivo, and this shows something. I mean, when we assess the phenotype and the functional capacity of the astrocytes in mice, for example, in rats, and the control with rats treated with mesenchymal cells. Once again, in order to support the principle according to which the mesenchymal cells act on other type of cells, this work has been done with Annalisa Buffo, and we took the inspiration from Magdalena Gross. The astrocyte can acquire phenotype, protection phenotype. So we show that the mesenchymal cells can transform astrocytes in scan cell-like cells. So those are cells similar to stem cells that can differentiate into neurons. So we have shown recently. So we kept on studying these issues together with the University of Bari. We have shown how the administration of stem cells can recover the functionality and through chemokine CCL2 and recover the functionality. So it is able to close the macroencephalic barrier, which has been damaged in the mice, and in humans. So I would like to close the section related to the mice, because I actually would like to tell you that the strength of our figures of our data is related to the immunitary system. But by the way, there are diseases, but in more diseases, the stem cells, the mesenchymal cells can improve the situation. Diseases such as stroke, for example, according to all. So amyotrophic lateral sclerosis, and the history is that the mesenchymal history is mesenchymal cells block TB and produce neuroprotections through some mechanism that should be that we have to underscore, because actually this is linked to the fact that this is done through bystander mechanism release or instructions of a mechanism that can act on other cells that are able to promote the recovering or reduce the damages. So actually, and there were initial studies pretty interesting because actually the administration, intravenous, intrathecal administration was meaningful. We don't know about the therapeutical level, but in 2010, thanks to the support given by the Italian Association, we founded the transnational group about stem cells, an awful lot, and in order to create the so-called famous MESM. And yesterday was talking my wife, and I was talking about it with my wife because actually the study has a specific so this here you have the goal, first and foremost, confirm the effectiveness, the security, and then assess the efficiency, because actually all the all the individuals should be treated and assessed. The number of patients and also the duration of the treatment and of the observation made it impossible, since we have those cells are really anti-inflammatory. We use the lesions by using contrast. So if those cells are really inflammatory, so they have to turn the inflammation off, and that's why we have. So there was a series of secondary outcomes, tertiary outcomes, and we are completing the analysis with a lot of problems. The study has been completed 2 years ago with a huge effort, with an awful lot of people involved. And at the end, we have treated more than 240 patients. So these are the demographics, which are pretty balanced. And here you have the result. The results are that there is no effect with respect to the placebo. We have taken into account an awful lot of secondary outcomes, and the information are true because according to all the analysis that we have made, obviously they are safe. We are aware about that. But patients don't want to be treated with something safe but not effective. So the treatment is safe, but the treatment is not effective. So there is a very last thing which is driven. So by the center of London. And this was the feeling that there was a hint of effectiveness in the, but actually effectiveness on ARR. These are all the studies that are not important, and all this. So I would like to thank the Italian Association for Multiple Sclerosis, and also I would like to thank an awful lot of partners that have collaborated. So you can see the number of enrolled patients. Italy has played a major role, but also we have to stress the contribution made by Denmark, Spain, for example. And here you have the name of the investigators that have been really important. And then, is there something going on in this field? Yes, there is something going on, because actually there are recent studies of the group of Dimitri Carusis, and also of other groups, Jeffrey Cohen, for example. And what can I say? Because actually the results are not conclusive, because actually the feeling is that there is a signal, but it's really difficult to assess it, because the other studies are not so huge, so big. So why MSC did not work or do not work? Maybe we will have a round table, we can discuss about it, but the source, bone marrow versus adipose or others, maybe manipulation of frozen versus fresh. Yes, of course. My friend Jacques Galipo says that the mesenchymal cells are like sushi, they have to be eaten fresh, and personally I agree with him, but unfortunately it was impossible to have a study like this without using frozen mesenchymal cells. There are studies saying that when those cells are unfrozen, the capacity drops. Route of the administration, maybe. So I don't think so. The doses, because actually we got started with a study with Caterina LeBlanc. I don't know, transforming what we give we give the mice into what we give the humans, so it's really difficult to make the difference. And also, for example, the frequency of dosing. So we administered once in 12 months, and maybe we should have given once in 3 months. The poor definition of cell products, because actually and other cells are produced in Sweden, the same things as the cells produced in Spain, and then the patient's selection. I think that this is the main reason we got started in 2012, and our target was patients with active disease. Meanwhile, an awful lot of drugs were released, and people wanted to do this treatment were people that were not active, and they had a low level of inflammatory activity, while all the other, as to the other active patients. So we should have had this treatment as well. Trial design, I end my speech saying that if we take into account the quotes of our work, that means that the basic science is credible, is reliable, and the number of quotes is really impressive. So I think that, and that means that nowadays people studying the biology of these cells think that those cells have a potential, but it didn't work. And this is contradictory in the journal. Mark Friedman and myself, we said that we were not convinced about these cells. So the results the results. So this is a study enrolling a huge number of patients, a study about on stem cells, preparation of cells, and the result is not good. But by the way, we have to be really aware of the commercial interests pushing towards a study, and this so pushing towards the treatment based on the need to give an answer, because actually we can't give an answer with drugs, but by the way with medications, but we have to take into account this issue with an awful lot of care, and this treatment should be done within clinical trials. Thank you.
Thanks to Antonio Uccelli for this interesting presentation. He rightly mentioned the efforts made in creating an international network when nobody had thought about it, and at a time in which it was really essential to provide information in scenario fake news, and I wish to underline this. But this enables us to learn, to know what happens in our body. And with specific reference to networks, a significant effort made by the community is exactly delving into specific studies with stem cells and hematopoietic stem cells in particular. It is an effort in which the Italian research is in the forefront. Therefore, I'd like to give the floor to Paolo Muraro, Director of the Neuroimmunology Unit of the Imperial College in London, to take the floor on hematopoietic stem cell therapy, for whom and when. Good morning. Thanks to Professor Mancardi. I'd like to start by thanking FISM and FISM for the support given to me, and particularly because they have helped me in particular for this research. We started with 30 Ore per la Vita in Canale 5 in 1995, and I had the opportunity of having my first scholarship at the NMH in Bethesda. And we started this collaboration relation that is still continuing. In my presentation, I will focus on hematopoietic stem cells. And I'm not just talking of this therapy as a monophase therapy, but as a therapy for the reinfusion of autologous stem cells. I do not know whether this is important. This is just a statement on my disclosure. But I'd like to start with thanks to a colleague, an hematologist, that after a long disease with cancer died last February. For those who do not know him and you want to have additional details on his career, there is an article, an obituary article in the Bone Marrow Transplantation of 2024, but I can provide some information on his significant contribution for patients with multiple sclerosis. I've here summarized information on his role. He was chairman of the working party of the self-immune disease of the EMBT, and he did a lot for transplantation in the field of self-immune diseases. And then he was for 30 years director of the unit in the Careggi Hospital in Florence that he is in the vanguard. And I wish to stress that Riccardo Saccardi made a significant scientific contribution, more than 210 index publications, of which 74 articles on multiple sclerosis. This is exactly the Van Beckham Award of the EMBT annual meeting, one of the most prestigious award on specific scientific goals. And as an hematologist, Riccardo is a typical example of the work done in a multidisciplinary team. Here we are in front of the famous Nuvola Conference Center here in Rome. He participated and was one of the most significant point of reference for collaboration in the field of neurologic sciences. So he really advanced this therapy for all the patients with multiple sclerosis. And this is the reason why I wanted to pay tribute to him, to Riccardo Saccardi. Thank you so much for this tribute paid to Riccardo. Let us now move to the structure of my presentation. The title is Therapy for Whom and When, but obviously we must better understand what this treatment is, how it is carried out in practice, because I want to tell you that it's not just an issue of hematopoietic stem cells. So there is a special section on self-immune disease. And I had the opportunity of explaining the principles of this therapy, bearing in mind that the self-immune diseases starts with the acquisition of a multifactorial genetic risk that is particularly complex, and that this risk alone is not enough to cause the disease. The disease takes place following a series of events during the life of the person and of factors. And obviously, the environmental factors are particularly important. In the case of multiple sclerosis, viruses and Epstein-Barr are significant. Therefore, all this system of factors is the system that causes the inflammatory process. In the case of multiple sclerosis and other self-immune diseases, this is a complex process, and the cause of this disease is sometimes not always understood. And it is really hard to understand the physiopathology and the etiology of this disease. For example, we have the problem of latency to these environmental agents, and also differences in the phenotypes of the disease. And these differences, this heterogeneity, makes things harder, but we can identify three main therapeutic areas in the field of multiple sclerosis. The first is stopping inflammation, and I believe that it is clear that stopping inflammation is useful in multiple sclerosis. And there are so much clear evidence that this can delay the progression of the disease and stop the loss of axons and neurons that then lead to this irreversible damage. There are treatments to stop the inflammation. That is obviously the essential phase of this disease. There are different strategies that were successful to different degrees. We have immunomodulant treatments, immunodepletive treatments. But, the therapy of the so-called immunitary reset is the therapy I'm now talking about. And there are some methods and principles that are particularly important. The autologous transplantation of hematopoietic stem cells is an example of imprecision medicine. This means that some specific treatments may be particularly targeted to a specific therapeutic goal, but in a context of pathogenic heterogeneity, cannot be effective in all different patients. So there are advantages because there are fewer off-target advantages, but the main disadvantage is they're not effective in all different patients. And it is an imprecise therapy because it is mainly based on the total ablation of the hemolymphopoietic system, and therefore it eradicates all the specificities, including the pathogenic ones, and has this great advantage of being more effective, considering also the disadvantages of a great level of toxicity and risks with a different profile compared to more tailored therapies. And, I like to stress the problem is not just the autologous hematopoietic stem cell transplantation to repair brain. Probably Professor Martino will certainly focus on this. But in this case, we have a multi-step procedure in which the central aspect for the efficacy of the therapy is the immunodepletion or immunoablation, namely you kill the lymphoid system with all the T and B lymphocytes that we all know are the mediators of the disease. And this process is carried out in a hospital setting, and over some weeks after the replenishment, at least initially, replenishment of the hemopoietic and immune systems, you can move from antibiotics and antiviral diseases that are administered in the hospital setting to enable disease-free life, avoiding daily or cyclical treatments, as is the case with patients that follow DMT therapies. This immunoablative protocol can be carried out in different ways, and obviously it envisages chemotherapy that is similar to the typical chemotherapy for cancer patients. And it can be carried out in different ways, at a different level of intensity, with the highest level of intensity. In the case of the ablation of the lymphosystem and the myeloid system, these are myeloablative therapies because they suppress the bone marrow, and they can be extremely effective, but obviously have a higher danger profile. And here, the protocols that are mainly used in this case are the so-called BIM ones and cyclophosphamide ATG. If we reduce intensity too much, at a certain point, you no longer provide hematopoietic cells, and the subject can recover the function alone, but there is a reduced efficacy. Hence, what can these hematopoietic stem cells can do? They help to repair the system, and this is a work carried out with Antonio 15 years ago. Though they tend to recover the hematopoietic system. These stem cells are taken from the bone marrow, frozen, and then infused again after chemotherapy. And obviously, this enables us to make the whole system start again like seeds in a garden. As to the mode of action, we can say, and here we have a summary slide, considering that obviously I cannot enter into details because time is pressing. There is a regeneration of the T cells, of the differentiation in thymus, that is the organ that promotes the differentiation. And then there is a regeneration of a new adaptive system, both of T cells and B cells, enabling to start without a cell load of the disease. Obviously, there is also the enhancement of regulatory aspects, especially in an early phase, enabling to reset the system so that it can, in a sense, facilitate immunological tolerance. This works very well, I would say. And just to give you a visual idea of what I mean, this is a phase of an American study, and it is one of the many examples because we have similar results. You have the relapses, disease progression, everything is in the phase before transplantation. And then you see that these events are spaced after transplantation. But there is something that we must stress. You know, in this study, we have considered 5 years and not for all patients. But what happens in the long term? What are the long-term outcomes after autologous hematopoietic stem cell transplantation? This is a one-off therapy, and unless needed, there is no long-term therapy. So what is the duration of this treatment? And once again, we go back to Professor Saccardi, because together with him, we coordinated a multicentric trial at what level, in which we collected information on 281 patients, if I remember rightly, with a long follow-up, average follow-up of 6-7 years. And so what are the long-term outcomes? We had different cases, but there are three main factors to be stressed. Age, so the sooner the better, so as to avoid the progression of the disease. Then the disease subtype, this is important because unfortunately the results are certainly better with remittent cases and worse in the cases of progressive disease. And another significant aspect, and I like to stress this because there is nothing we can do in the case of age, for example, or the subtype. But in the case of therapy, we see that the results are worse in the patients that before transplantation were administered different lines of therapy, because this shows that multiple failure of therapies, also the transplantation has a very limited effect. Just to give you a scale of efficacy comparing the current therapies, in this study carried out by Riccardo Saccardi, we considered the results of the DMT therapies, including the current ones, for example, oprelizumab. 50% of patients without evidence of disease at a follow-up of 2 years. In the cases of transplantation, the results show a rate of NEDA higher than 70%. And this is seen also in studies carried out after 2017. But obviously, this is not trial data or comparison data. This is just a compilation of data. And therefore, moving to randomized double-blind trials compared to standard therapies. There is a first study led by Professor Mancardi that, needless to say, was also a close aid of Riccardo Saccardi. And together they carried out this Hastings study, a small study that compared patients that underwent transplantation. And we had magnetic resonance tests. And we have realized that the problem is mitosantrone that now is no longer used. And therefore, there is another study, the so-called randomized control study, RCT, led by Chicago, Dr. Berth, that showed a significant superiority of transplantation compared to control therapies in a wider range of patients that mainly carried out first-line therapies, interferon, for example. Only few people were administered rumatalibulab. But it is not easy with this study just to compare these different therapies. And once again, Riccardo Saccardi sets in. In 2012, we promoted together a position paper that designed the vision of how to organize trials to assess transplantation. And basically, the principle of this trial is that transplantation must be compared with the best available therapies, because otherwise, mitoxantrone, for example, has better effects. But when it is no longer used, obviously, we need to somehow make all this trial topical. And therefore, we organized other studies. There is one that is almost completely RAM-MS, a Scandinavian and Dutch study. Then we have BDMS and STAR-MS. They have reached 2/3 of patient enrollment. They are mainly American and British studies. COST, unfortunately, was terminated because it was not possible to enroll patients. It was a German COST. And then we have the NET-MS Italian study that is now led by Professor Matilde Ingles, and we hope that we can start as soon as possible. Finally, the last part of my presentation focuses on the patients that are candidate for this treatment. And we must be very clear, because even though we would like to provide greater help to patients with advanced type of multiple sclerosis, unfortunately, evidence shows that in the advanced stage of the disease, the risk of transplantation regarding immunosuppression, heart toxicity, it is anyway chemotherapy, and therefore the risk of this part of treatment outweighs possible benefits. In the framework of the early and remitted and exacerbating forms of the disease, as well as the early progressive phases, the potential benefit is more marked. Hence, considering therapeutic positioning in an appropriate context of experience and expertise, transplantation can be proposed as a standard care with progression of the disease when there was a failure of the first high-efficacy treatment. I do not believe that all these treatments must be highly effective, because otherwise you lose the window of opportunity. In particularly malign and aggressive forms, you can use this treatment as a first line. This was carried out with excellent results. But, only in selected cases by experienced centers. We can consider also the active forms of multiple sclerosis with inflammatory activity. So this type of patients are those who most benefit from this type of treatment. There are some guidelines of the MBT, but Italy, also thanks to the efforts made by SIN, as some guidelines of the Higher Health Institute stating that selected cases not responding to high-efficacy therapy can be considered as candidate for transplantation in centers that are particularly experienced in this procedure, and therefore including and considering the risks and the benefits of the treatment. This is essential, and therefore we need to have a collaboration not just between the hematologists and the neurologists, but also between the patients and this team of practitioners, because we need to have a clear understanding of all the aspects of the therapy. I wish to thank my aides, my team, working in various centers. And I'd like to say that the Center of Hematology, led by Saccardi, will certainly continue. It is in good hands, and we will define this in the near future. But certainly the contribution of Riccardo will never be forgotten or given up.
Thank you. Thank you, Paolo. We will have the discussion during the round table. Gian Vito Martino, who is the scientific director of San Raffaele. He worked a lot on the neural stem cells, and he will talk about neural stem cells that could revolutionize regenerative medicine. Thank you very much. Thank you for this kind of invitation. I will be short in my speech, but basically I would like to say something about experimentation, and would like to tell you the 4 stories linked to stem cells and how they contribute to changing the therapy, and how they give us some important opportunities. The main thing, I don't want to dwell on this, but by the way, we should say that the multiple sclerosis concerns brain, bone marrow, and it's a multifocal disease, pretty complicated, complex disease, and it's not easy to carve out the therapy able to fight against the inflammation and myelin depletion. So from a therapeutical point of view, this is a problem. This is a huge problem that we have to face. So treatments were based on prevention. I would like to say that Italy made a huge contribution since the very beginning, because actually the idea of having having treatment, immunomodulation treatment, and according to the data, we can draw these evidences. And by the way, we have said that the autoimmune process characterizes this disease. Some 20 years ago, someone said that the progression was totally different, but we always said that this disease was simple. This disease was a consequence of the autoimmune factor, the bone marrow, the treatment of so we know the cause of this disease. So the first chunk, the first part is related to the clinical experimentation that we carried out with cases you see 2003 mice, mouse, 2009 human fetal neuron stem cells, 2017 human fetal neural stem cells. So we have the best standard effect, and actually it has been shown with in 2000 more or less, and with a neural and then mesenchymal stem cells have shown that a part of the effect is not to substitute, is not a replacement of damaged cells, but actually a part of the effect is related to the secretion of molecules, neuroprotective molecules. And I think that this data, these figures have paved the way to all we have now, vesicles, secretomes, because actually this, for the first time, we have shown that these substances can regenerate. And the trial has come to an end. We have published the data, 12 patients. I would like to thank Angela, Milena, Francesca. And actually, this trial was really simple to do, easy to do. It's far more complicated with respect to the pre-trial activities, the regulatory activity that we have to perform. And we had injection on 12 patients with scalable doses of neural stem cells, 50 million, 500 million, and then we assess the safety. The therapy is pretty, the transplant is pretty safe. But the most interesting thing is something which was totally unexpected, and the following. Totally unexpected. The more you transplant stem cells, the less the disease progress. So the data is this data is pretty surprising, and needless to say that we have an issue. We had an issue with the small number of patients with respect to the quality of data. But by the way, the data that I will show you, which is a long-term follow-up, are really challenging, because after 6 years from the transplant, in the 2 following years, there was a less depletion of gray
sulla dose più bassa, diciamo.
Of gray matter, sorry. And also there was an indication, and actually we are treating the processing the data. I would like to thank Dr. Rocca. We are analyzing and processing the data, but by the way, a correlation may exist with the improvement of the cognitive activities. I would like to dwell on this because I think that this is the most important part, because actually when we did the proteomics on the transplanted patients, we saw that the stimulation was concentrated on the axons, and so as if myelin had forgotten
what the heat was doing, and it was concentrated on the somal damage. Obviously, the following step was to do we conceived a phase 2 trial, which is really complicated because there are this is a multicentric trial. We have a limit of some hours, of 2 hours from the production to the administration of the cells. We had to involve only colleagues near us. In Lombardy, we have spoken, so all the centers of the multiple sclerosis in Italy will be involved when we will have the authorization, the green light, and also they can choose, the centers can choose patients. So we once a month ago we have asked IFA the authorization for the phase 2 of this trial, and I think that we will have a green light. So we do hope to have the green light by the end of the year. So this is the procedure just to scare you, because actually the paradox, it's weird, because they ask us to behave as a multinational company, and they don't understand that what we can do is totally different with respect to a multinational, and because we need support in order to boost the development of our research as a university. And so in a certain way the regulatory authorities should understand how these therapies can reach the market. Having said that, let's have a look at the future and try to understand what will happen next. Obviously, the limit of our study is the use of autologous cells, so we have to do transplant, and this is really complicated because there are a lot of drugs that have to be administered, such as antipsychotic, and this is sometimes difficult for people who have walking problems. So we have these cells, pluripotent stem cells. This is the novelty of the moment, and now they are available. They can be used. They can be taken from the patient, so autologous pluripotent stem cells. We are working on this, and we have there are some guidelines. There is also a ministerial project funded by the Resilience and Recovery Plan in order to research on this. Doing research about these cells. Obviously, the first thing was to understand if these cells can be derived from the patient, and if these are normal cells, because this disease affects the neurons. So, for example, maybe the risk is to have cells which are affected by the disease. So these are derived. So we have derived the stem cells from patients, and then we have tried to understand if the cells were normal. In order to do this, we have used cells coming from monozygotic twins, and we saw that those are the cells are the same. So in these twins, the proteic expression is the same, and then we have created a model which is really interesting.
which is with a human myelin, so a complex model, and we saw that the cells, even in a functional setting, can produce a good myelin, even if those cells are derived from a patient affected by MS. So we have also oligodendrocytes, which is not the only target of this disease. So this disease is a global, a general one. But by the way, these cells are not changed inside, are not modified inside. So and then we did a transplant. We have transplanted these cells in animal cells. So obviously, all these studies are preliminary studies in order to allowing us to use those cells for our therapies, which is the ideal solution. But by the way, neural stem cells can be used for different things, and not only for being a therapy, but also in order to be the the way we can reach a new therapy. For example, this project in which Italy is involved has come to an end. This is an international study. We had the pleasure to coordinate this study, and it's a multicentric study trial. We got started from 1,500 molecules adopted by the FDA for human use. We used a system called Spoke. This is a database in order to filter in those drugs, in order to understand if they are related to the disease, and from 1,500 molecules, so we have kept 2 molecules, and there is one molecule which is pretty interesting, which is an antagonist of H3. So, but beyond the results, you can see that in the pipeline of assays that we have put in place, you can see that an awful lot of guidelines are based on stem cells and neurons coming from patients, and also you can see on your right the three-dimensional structures, mini brains we call them, and they can be obtained and studied only thanks to the stem cells, and the stem cells are a source of cells that can be functional cells. Paola has followed this work together with Svetlana. And this Bavicent. Bavicent is a molecule which has been used for sleep diseases and sleep disorders. This cell is neuroprotective, and actually the principle was to find a molecule having both activities, both functions. So we keep on working. We are going to deliver an update on this project, but by the way, the idea is that this category of drugs may have a dual effect, so they can be able to, and they will be able to be part of the clinical activity in order to be used in the phase 2 trials, effectiveness trials. But coordination is really so. There is something interesting with respect to stem cells and multiple sclerosis, because actually, as you know very well, the stem cells that we use in the brain are around the ventricles, in the zones in which you have more lesions before of this because of the disease. This area is intertwined with the striata which is the only brain structures able to send electric signals. So the striato is a sort of smart filter deciding which are the messages that have to be brought. Since those cells in the brain are normal and they are closer to this area, so in a certain way they was related, and we thought about explaining why they have a kind of behavior in vivo. So and then in a laboratory, we have generated an awful lot of years ago, more than 12 years ago, we had created a rat in which we were able to eliminate the stem cells. We were not following this, but we have recovered some data, and we understood that those rats were not able to make a decision without the stem cells. Their phenotype was more normal, but it was really difficult for them to make a decision. And to cut a long story short, so we took into account at this point, we took into account the patients affected with multiple sclerosis, with the damage in the ventricles, and we saw that there is a correlation between the atrophy of the subventricular zone, the striatum, and the alteration of the cognitive functions. And then we tried to give an explanation. More than 60 patients affected with multiple sclerosis, and we saw that this is related to the fact that these cells produce EGFPPL1, which is a protein linking a trophic factor and make it available to its receptor within the striatum. That means that there is a trophic process, because if there is no continuous message, the cells in the striatum does not do its work. So without those neurons, there is a loss of connectivity, there is no inhibition, so the brain is not able to make a decision, to think properly, because the brain is not able to make all the mechanism, bring all the mechanism together. So I don't have the data now, but according to some indications, by changing this pathway, we can revert the phenomenon, because this protein is able to give the functionality back to the neurons. So we can say that this is maybe one of the mechanisms when we do the transplant of stem cells. There is an effect which is clear on the cognitive functions. Actually, so this work will be published soon. We have transplanted these cells with a severe neurological disease, and in this model there is a protection of cognitive functions. So if we think about the fact that we are going through, so we have to take into account the cortex and all that we have to do. We know the role of neurons in these functions. So in a certain way, so I have to end my speech because we are running late, really late. So according to to the indications given by our president, I leave it there. Just a short conclusion to tell you that the stem cells go beyond the transplantation, and it's a tool allowing us to study biology, a very interesting, riveting tool in order to study our brain. So even if we started from the clinical to the biology, actually we are discovering information that hopefully can be used also for persons, humans. So so there is a very last thing. This kind of project without a foundation such as the one that welcome us today would not be possible, because those projects entail an awful lot of risks. So we have to believe on those projects. The foundation believes in these projects. So we have been working together for ages, and I would like to thank all the staff of FISM. Thank you.
Grazie. Grazie Gianvito.
Thank you, Gianvito. I mean, you did not comply with the time allotted to me, all of three, but you have provided a clear framework for this issue. But now we are about to start the institutional event. For those who are connected now online, and the institutional part of this event is a way to focus on these issues and advocacy, care, and so on and so forth. But now we would like to take stock of the situation and the setting in which we are operating. For example, the importance of the non-profit funding for scientific research. So now I'd like to give the floor to Gabriele Beccaria from La Stampa, JD Group, that will coordinate this roundtable discussion. Thank you, Professor Battaglia. Thank you to all of you for the participation in this conference. In the second part of this morning session, we thought to organize a roundtable discussion focused on the issue of clinical experimentation and advanced therapies. Explained by Gianvito Marteno, Antonio Uccelli, and Paolo Muraro. As already said, when we organize a roundtable discussion, you need to involve many different people playing different roles, and therefore the so-called stakeholders. So today, we decided to involve many different people with different roles, with different skills, to try and give you an idea of this disease, multiple sclerosis, not just as an individual tragedy, but as a collective problem and as a real universe in which many different aspects set in. So we can use this concept of complexity, many different elements that interact and lead to significant and variable results. Hence, I like to invite here the guests for this part of the session: Gianluca Pedicini, that is the president of the Conference of People with Multiple Sclerosis from FISM, and Angela Martino, former president of this conference. Then we have Mario Alberto Battaglia, president of the FISM Foundation, the Foundation for Multiple Sclerosis. Then we should have had also Francesco Saverio Mennini, the head of Department of the Health Ministry, but unfortunately he could not come for an unforeseen engagement. We have Sandra Petraglia, Director of the pre-authorization area of AIFA. Then we have Mario Zibellini, Technical and Scientific Director of Farmindustria. And then, last but not least, the three neurologists and researchers took the floor before me, namely Gianvito Martini from Raffaele Milano, Antonio Uccelli, Scientific Director of the San Martino Hospital, and Paolo Muraro from the Imperial College of London. So welcome to all of you. So as you can see, we are here with many colleagues, and our idea today is to try and see this disease from many different viewpoints, trying to have a holistic approach that is a specific characteristics of medicine of the 21st century, in which you have different elements that interact one another. And considering that we have basically 1 hour to talk about these issues, I'd like to ask our goals to speak roughly 4 or 5 minutes, and then, if possible, we may have a second round of contributions. Because we would also try to provide some suggestions, possible solutions, considering that we are talking about many issues that we are tackling, but that also needs creativity in order to find the right solution that may turn multiple sclerosis into a real case study, a disease that can set a model also for other diseases and for personalized experimental and technological medicine. That is the typical medicine of the 21st century. So I'd like to give the floor to Gianluca Pedicini, because I think that you somehow represent the new type of patient that is not just a passive patient to whom treatments are administered. Patients are active patients. They can contribute with their experience to refine treatment therapy, and therefore this is extremely important. Good morning to all of you. I like to say that if our goal is that of creating responsible health and a sustainable model of healthcare, we certainly need to involve the patients that are affected by this disease. Our association is involved in many Italian and international projects based on this multi-act model, in which there is not just the single person that is involved, but a real community study. And this community study leads to say that the experience of the life of the patients is a heritage of this research. And on the other side, this gives data that are not just data related to individual patients, but to a community. And therefore, if these data and information are provided properly, they can provide useful indications. And probably this was just the missing point of research so far. And probably these indications are not provided only by the people, but also by research. You know, now time has come to start this process. Considering that we are talking about this notion of community, of the involvement of people, I'd like to ask Angela Martino what this means in practical terms, because we know that communication has a fundamental role, especially at a time when, unfortunately, scientific and medical information in particular is contradictory. It is fairly good for some aspects, but very weak for others. And, now fake news are daily practice, and especially when we talk about multiple sclerosis, many people have improper ideas. They mistaken this disease with other diseases. So we are faced with people that on the one side they want to know, and on the other side are flooded with contradictory and fake news. So the situation is constantly changing, and therefore, how can we make things clearer without oversimplifying things? Because, medicine and research teach us that we must see things as they are, a complex process over time. Yes, that's true. We should not necessarily simplify it, but make information accessible, because this is essential, especially when we talk about scientific information. We all know that in Italy, we have room for improvement in terms of scientific culture. I believe that the post-COVID events showed this. From my viewpoint, a person with multiple sclerosis, information is a therapy. It is therapy because when I know the benefits, the risks of an innovative treatment, or when I know that that innovative treatment that has not been validated yet, but that could be extremely useful for a large group of patients, I believe that we have the right, but also the duty, to provide correct information, accurate information. And I'm talking about right because as early as 2014 in our Charter of Rights, we included the right to proper information, to accurate information. And I wish to raise this issue on the importance of being promoters of accurate information at all different levels. Multiple sclerosis patients, but also researchers and institutions on the other, because what we are talking about is the importance of data and information. The fact that they can realize whether an innovative therapy has become therapy and has the possibility of providing a concrete impact on the life of the people, because otherwise those data and information remains sterile data. So I believe that we can create a virtuous cycle. But now I'd like to give the floor to the neurologist so that we can better enter into the details of the issue. Thank you. So considering that we are talking about virtuous cycle, I think that we should put together all the parts of this picture. So I can ask Mario Battaglia what is and will be the role of non-profit organizations. Basically, the two most familiar words in this respect are advocacy and engagement. The information of the associations of patients like yours is of fundamental importance to promote the different aspects. So it is a work in progress that is playing an ever-increasing role, not just in the field of multiple sclerosis, but also in other diseases, rare diseases, and ADS, for example, in the past. So how do you think you can further develop this commitment that has been characterizing you for years now and is universally recognized? You know, the commitment must be made on different fronts. The easiest one is probably just funding of research, because as already said, believing in research means that you should do so also when there are risks, and this is a task that can be performed basically by associations and foundations like ours rather than other players of the system, including pharmaceutical companies. Therefore, our task is to continue ever more to fund basic research, to reach the so-called death valley in which it is necessary to achieve the real development of drugs. That is not obviously our task, but of pharmaceutical companies. And, you know, we must be protagonists of research. And this means, as already stressed, to be able, as association, as people, to guide, to orient research, because this is not just an issue of funding. We've already mentioned the example of stem cells. There is this need for trusting stem cells and invest in that research area. When we started to fund stem cells research, it was not so easy. It was 20 years ago. But the most important thing for our associations is to promote, create networks in which all the different players can play their roles. I'm thinking about the activity carried out to promote the network of neurologists, clinicians, basic researchers, and so on and so forth, as mentioned in the previous contributions. But there is also another phase that is creating, designing clinical trials that are not the usual heritage of those who invest in clinical research. And, you know, funding these trials is certainly significant. As already mentioned, for example, we have phase 1, phase 2 trials that already require a network to be established and not just funded to reach significant results. We also have examples, and indeed we have raised also a problem in the previous contributions. If we demonstrated that a certain product, stem cells, for example, can change the life of the people, who has to invest and formally produce and distribute the stem cells in the system? You know, this is a fundamental issue, because in the future, as well as current scenario, the associations may play a fundamental role in the system, obviously a non-profit player, but that can support and invest in the network to provide responses. For example, mention was made of rare diseases. When we talk about therapies that are only for a specific group of people, we need to have a holistic approach. It is hard, I know, but I think that we must take our responsibility as associations. Hence, we are talking about networks and establishing networks, as well as networking, so to say. And I want to ask Sandra Petraglia what this means, because we know that one thing is imagining these networks, but then we are faced with advanced therapies, and then we have also different needs, treatment needs for individual people, and then the need to define rules that can be recognized at universal level. And therefore, we need to define new models, regulatory and organizational models, enabling us to put all these different elements together so that they can make these therapies work well for patients. First and foremost, I wish to thank for the kind invitation. You know, networking. We have talked about rare disease and drugs for these diseases. These are tailored and personalized therapies, but today we are talking about multiple sclerosis, and therefore they are for pathological conditions that are ever more widespread. Therefore, I believe that we are moving towards a scenario of greater spreading of these therapies that are particularly complex due to basic research, discoveries, and then there is also the productive aspects. The higher the number of patients, the greater the complexity in a specific set of rules and regulations that should be standardized in terms of criteria. You know, the efforts made by the regulatory authorities and of the health ministry, those who have to set these rules at European and world level, realize that this should reflect also the organizational aspects when these advanced therapies can become a real reality of treatment for these patients. So we want to have this organizational aspect that is based on healthcare. What we are witnessing is the shift from niche diseases to a more diversified disease. We may be faced with a scenario in which the specialist is not necessarily in the center for the provision of these therapies, of these innovative therapies. Hence, we must rethink the organizational criteria for experimentation, interaction between centers, and for the administration of drugs to patients and the follow-up, as well as the better distribution equity from the geographical viewpoint, because as you may know now, we have more therapeutic options focused on some geographical areas compared to others.
So, starting from a dialogue, a relationship with the clinical institutions or clinical centers, so we would like to carve out a model leading to a structure, affordable and reliable and sustainable structure. So we have to work together in order to standardize the customized approaches, in order to streamline costs, and in order to have affordable therapies. So the studies are more than 100 worldwide, and by 2030 we will have more or less 50 authorized therapies. We have to set the standards. So IFA has to define the criteria in order to have the accreditation of the centers, and I would like to seize this opportunity in order to tell you that the approach is not on limiting things. We have to ensure quality and safety for all patients. So requirements should not hinder the development of new products. So the dialogue with the stakeholders is really important in order to carve out the rules, keeping reliable for good standards. But the idea is not to hinder research, new products. So I think that from this standpoint, I think that the pathway will be. I think that we will definitely come down this avenue. So Marco Zibellini, since today we are putting together some words, costs and sustainability, for example, and I think that for the pharmaceutical industry, those words are really crucial. We know that those therapies are complex, are complicated, time-consuming, and also those therapies are not easy to fund for the industry. So they entail a structure, an organization able to ensure this mechanism going from the production to the administration to the treatment can work. So I think that this is a crucial issue relating concerning all the patients, not only affected by MS but by other diseases. I'm thinking. How can we work in this field? Because, by the way, we have achieved some good results so far, but once again, we are talking about a process, an ongoing process, which has not come to an end. Thank you for your kind invitation. It's really useful to be here because we can have a fruitful discussion with all the actors, all the stakeholders. So this discussion, this debate is crucial, namely when we deal with advanced therapies based on cells and on genes, which are the new frontier. The industry is investing massively, not only the industry, because in 2023, 631 studies started and 36 of which industrial based, and so today we have witnessed an awful lot of initiatives. During the last report, we have been we got started from the clinics, and then we go to biology. So and this, so there is a preclinical phase, clinical phase, a productive phase. So those trials are pretty complicated, and also how to deliver those therapies to our patients. This is crucial. Given the complexity of these therapies, we said that we have to work together. Because actually, if we work together, if we pull together all the experts, the scientific experts, the industry, the regulatory bodies, we can streamline the procedures that are really important in developing new therapies. And along the pathway, we have to adjust and find solutions with a view of streamlining and supporting what we produce, because there is an awful lot of fake news. We think that it's really simple, easy to administer cells but this is not the case. So often, so we spoke about the new technology. So, and people say, well, it's really easy. We can produce this in Italy, but in order to have these products, these drugs in Italy, there are problems because we need the quality, we need the right structures to do this. So I think that, and also as far as the research funds, so the funds allocated to research are really important. Italy is behind China, for example, in attracting industrial investments, and also, so this is something that we have to do together, because I think that we have, it's really important to pull together institutions, experts, industry, patients, in order to work together, because this is important in order to achieve tangible results. So we have been speaking about food sustainability, financial sustainability. It's really important to have an industrial system and to ensure the return on investment in the long run. The clinical development is really complicated for some diseases that have different phases. So in some phase, maybe in a specific phase, we can use stem cells, mesenchymal stem cells, or other type of cells. So it's really complicated and the amount, the money needed for these therapies from a commercial point of view is pretty. The price is really high. So we have to understand how the NHS can ensure this, can pay these costs, and since the results are in the long term, so we have to improve the production phase, the preclinical phase, and the regulatory bodies can help us in establishing customized pathways for these therapy for these diseases. But I think that in the future we will have a central European assessment, because all the countries will be not left alone, and there will be something in common, and how to define the value of the new drugs, and we will get started from the advanced therapies in 2025. Assessments, centralized European assessments that will be evaluated at the national level in order to make a decision, in order to allow the access to those therapies. Sandra, in Italy we have a national health system, but regional health systems are really important. So we have 21 NHS in Italy. So we have to take into account the complexity of our administration, and we have to establish a link with the decision making at the national level. And it's really important to work together. Thank you. Thank you. So I would like to give the floor to the researchers that took spoke this morning. I would like to get started with Professor Martino. So I think that we have been impressed by the fact that the researches that have been explained are the result of a long and difficult work. It's a multidisciplinary approach, and during the morning session, I was thinking about the cases of huge spatial projects. When NASA conceived the projects related to exploration in Jupiter or Mars, for example. So when we see those spatial projects, we are faced with extraordinary visions entailing huge teams and long time. And often there are scientists embarking in this adventure as students, and along their career they keep on working on these projects, and they don't witness the end of the project. So I think that you are. So I'm sorry, I was exaggerating a little bit, but by the way, these researches are pretty long, pretty complicated, and then there are results that disclose further problems, further solutions. So those researches are very difficult to understand by the public. Yes, this is the case because actually I would like to thank Dr. Petraglia for this opening by IFA with respect to the advanced therapies. We have been working together for the genic therapy because actually we were both not aware about what we were talking about. When we speak about innovation, before innovate, you have to understand and know what you are innovating. So there are things that the industry did not do because obviously the industry is always always has some difficulties in facing the unknown. I have to recognize the complexity of some therapies, but there are an awful lot of systems that can be used in order to keep on working on those therapies. I think that the industry is not aware about the importance of advanced therapies. I understand that it's easier to have to create a chemical molecule. It's easy, but needless to say that the perception of the strength of advanced therapies is related to the scientific part, and this allows you to understand that this is the way with respect to drugs. So we are in the imprecision field because out of 10 drugs in the market are effective for 50% in the patients. So the advanced medicine has the hopes to be more effective and more precise for the patient, and the partnership between the industry, the university, the regulatory bodies is crucial because the science is not something a standalone thing. The science should work together with other stakeholders because we have to protect the health of people. And so the problems related to profits, investments, return on investment is a secondary and ancillary problem because what I have to do is to work on something in order to achieve results, and those results have to be achieved for the community, for the patients. So from my standpoint, and in order to answer your question, which is the best way to support the work of researchers, this is not something that we have to invent. In 2008, a book, Triple Helix, it's a crucial book about speaking about dealing with innovation, and it was about pulling together all the stakeholders. It was a triple helix, and this triple helix now it's quadruple Alice, because we have to involve other stakeholders beyond the experts, university, government, and so on and so forth. So I think that the Academy should create drugs, low-cost drugs, and this is something happening at the European level, because actually this is something that you can do. You can, and these advanced therapies can be affordable. There is a project foreseeing 3 hubs in Italy, and in the center and the south and the north of Italy, and those hubs are creating factories in which we can run this type of activity. So and also there is a problem related to the so the costs of a product created in laboratory are really limited. So we have to it's really important to take up our responsibilities when it comes to genic therapies. You know that we have the first therapy adopted by EMA. So the cost is 500 euros, and the Teleton, this therapy was not interesting from a commercial point of view. The Teleton Foundation tried to market, to put this therapy in the market. But so I think that there is no other options. The only option to decrease the prices was so I think that we have to understand that we are not talking about solar screens. We are talking about therapies in order to save children, for example. And this therapy and the therapies and the costs are really high. So when the costs are high, what should we do? Not using the therapies because of the cost, and at the same time we are not saving lives. I think that we have to face this issue, this problem. Really. So the Academy is ready to do this, and actually all the works that we have been doing in the stem cells field have been possible thanks to the FISM, and the results could not have been achieved. So why? And it's really interesting because we are always talking about the capital risk capital, but actually the risks are limited because you have there is no personal interest by as far as we are concerned. So there are some important books and all the books say that, and there is a system. For example, we have a phase 2 without the companies, without businesses. We can do this cell-based therapy phase 2 without the companies. So this is something possible. Yes, this is possible. Obviously, if we have a phase 3, we will need a partnership. But maybe we should we should work together since the beginning. We have to pull together all the competencies, all the skills in order to avoid mistakes, because this is the knowledge-based medicine, and then the economic side is important. But by the way, the economic side has to support the knowledge-based medicine. I don't know if I have understood I have answered your question. Well, thank you very much. So I think that Gianvito Martina has been really clear. Since we are in a round table, I think that we so according in my personal I have been working in a department of research and development, of a company. And so I take your point because actually at the end, this experience stems from a partnership between university and industry, because actually it was really important to have a scaling up. So starting from a therapy developed in a clinical center with all the knowledge which is needed. So
assolutamente essenziale.
When we talk about open innovation, this is essential, but this is another issue because, we have different cultures, and making the therapy that was already effective for some patients and use this for all patients in Europe, it took 5 years to define the regulatory dossier. And so this is an important problem. And the therapy had already been a functioning therapy. It worked well on patients. That's the reason why I decided to mention this. But this is important because I believe that we should agree on this. I know the problems because obviously we also had logistical problems because obviously the European Medicine Agency told me, okay, you do this in Modena, for example, but then we have to transfer these cells to London. Cells must be preserved in good shape and good quality. So I need to have a structure that is ready with the resuscitation unit and the patients ready to wait for that therapy in 36 hours. But what happens if there is a strike, if I have, for example, a problem with the channel? Otherwise, we waste these cells. So I fully share what you said. When we use the word cost effective, this must be assessed because, it is extremely interesting to put this in place. For example, I was talking with a colleague on the development of a specific drug in the United States. And clearly, when we develop, for example, the cells in the Bambino Gesù Hospital, we treat our patients. But obviously, we need to create other productive plans in order to improve the situation. And I believe that it is extremely important to stress this. And this is the problematic issue, as you mentioned. The problem is that, for example, the first therapies that have been marketed in Italy are no longer marketed. Now everything is developed abroad. Teleton has approved the therapies, but we wasted the heritage of therapies developed in Italy that are no longer at an industrial level. And therefore, these are issues that can be solved only when we sit together at the same negotiating table. So I have some experience in this respect, and I know also the complexity of the problem. The European Medicines Agency asked for having a specific clinical study. And also the issue of the so-called out-of-specific patches. This is another important issue. And the regulatory authorities tell me this is the registration, this is the price, but what we lose is material, tissue. So there are so many technical aspects to be considered. These are live cells, and there are different types of cells, for example. And I know, for example, that there are specific channels in which I have to channel and to inject these cells. And, the regulatory authorities, I believe, are sensitive to these issues, and they're also sensitive to the quality of data. These drugs have not obtained clear and full authorization for the drugs that are not reimbursed by the National Health Service. So the structures, the hospital structures, have to tackle these issues without a specific dedicated budget. And so once again, we need to tackle the problem of the funds for research. And I wish to reiterate that also involving the patients is essential. And these patients must be experts. And the experience is not just coexisting with this disease. All these processes must be assessed in order to be more incisive in terms of initiatives by the regulatory authorities. Indeed, this is a roundtable discussion, and therefore this is exactly what we are here for. Antonio Celli, I'd like to have your opinion, because we are ever more understanding to what extent the work of researchers is difficult. That must be seen in a specific network. We have also this issue of the virtual networks, the RIN networks. So there are so many aspects that come into play. With specific reference to the final outcome, I fully share what was said by the previous colleague. I believe that sustainability can be ensured only through some partnerships with the various players and stakeholders. As to the other things act, I do not always fully share these views. I can say from this viewpoint that what has been said today and to the results of excellent research, especially in Italy, there are no doubts. So I think that the idea of outcomes of the research is excellent, and the role of the associations for multiple sclerosis has been significant. Then we reach a stage in which, if we talk about cell factories, over the last 15, 20 years, we can say that they are increased in number. And if we consider the sustainability and the efficacy of their contribution, honestly, we should know to what extent these factories work. For example, in the Liguria region, now we have 2 cell factories. And then sometimes we realize that they have not sufficient work to justify this number. As Gianvito said, this is not just a problem of public academy. I am a university professor and I manage a public center. You mentioned, for example, the national centers, because we have millions of money from the National Recovery and Resilience Plan. But then what will happen in 3 years? How can we manage all these systems, all these factories that we have created thanks to this money, without having always clear ideas? This may be politically incorrect, but I believe that we have the national centers, the partnerships, But then all these centers must be made sustainable for the future when this money is no longer there. I may have some ideas on possible solutions, but I believe that nobody can solve this problem on its own. And I'm saying as much because, I'm here as a researcher, but I also play another role, as Gianvito said, and rightly so. This is not my problem or just a problem of the person with multiple sclerosis. Because when I am leading a public center and I see that obviously the budget is to be divided by the various hospitals, the problem is that I have to carry out spending review. And we have a lot of agencies that check, monitor, audit our processes. So the issue is complex, and there is no one solution that can fit to every situation. I believe that the only way is that we must sit together with the 4 or 5 significant institutions that can play a role in this respect, because we know that we have budgetary constraints. While we know that the public entities must protect patients with multiple sclerosis and have limited resources, the industry has certainly more resources. But we must be very clear. Investment is done if there is profitable. Otherwise, I prefer to leave everything to the public hands. So we must be very honest in our approach. We must realize that certainly the mission of doctors and researchers is to treat and cure people, providing the best possible therapies. And multiple sclerosis is an example of the importance of treatment and health care in our country. Probably most has been done in the field of neurosciences, and it is a disease that, thanks to the studies of the international community and thanks to the Italian Foundation of Multiple Sclerosis, much progress has been carried out, and we have responses to this disease that were virtually inconceivable 30 years ago. But now we must be honest, and we must know what we can do and what we can do for our patients. Because sometimes all the different players want different things. And so we must find an agreement in this respect. Paolo Murano, you'll have the task of drawing the conclusions of this roundtable discussion. Thank you. I have the advantage of having listened to the whole of the discussion, and I'd like to go back to some important issues. First and foremost, focusing on what was said by Angelo Martino, I'd like to say something with respect to information. On the one side, the supply of information affects demand. So, while we have TV programs that are detrimental to the culture and knowledge, this is a vicious cycle. Obviously, we cannot check the consumers of this information because patients with multiple sclerosis obviously look for information when they can find information, but we can check the supply of information. For example, in the case of stem cell therapies, if the information is easy to be found, we can, in a sense, prevent people from getting information on sources that disseminate fake news. Hence, from this viewpoint, I believe that FISM plays a fundamental role in proposing quality information. We can do better, I think. For example, we can enhance collaboration with the centers such as GITMO for bone marrow transplantation, for example. They also tackle issues such as the advanced cell therapy, such as the CAR-T therapy. So basically, we have information channels where you can find high-quality information, so we must promote them to get reliable information. A second issue on which we can debate is the cost. We touched the issue of the sustainability of therapy for multiple sclerosis. And on the one side, for example, Gianvito spoke of how we can introduce low-cost drugs. And I'd like to clarify that on the one side, we already have low-cost drugs, generic drugs, and bioequivalents. And this may be important for meeting the specific requirements of public spending. On the other side, as to the transplantation, and in order to better clarify the situation, because, I emphasize the complexity of these therapies, but this is a therapy that is available that in the UK, in Italy, indeed costs roughly 50,000 euros. And it is therefore a potential source of economic saving compared to biological products. For example, we spoke of the complexity of funding research with advanced therapies. So on the one side, focusing on what was said by Antonio, Antonio mentioned that the academic world can do miracles in designing a low-cost clinical trial compared to magnitude of this endeavor. And I can correct you, Antonio. MESMS indeed worked because it met the scientific demand. And obviously, the trials are carried out because we want to check whether a therapy works or not. Moving to the issue of sustainability, there are opportunities for increasing the sustainability of multiple sclerosis therapies. Obviously, we provocatorily mention also transplantation as an imprecision therapy, which is not really the case. But, this is the principle. how can we see this with respect to precision medicine and tailored and personalized medicine? Because moving back to the patients with multiple sclerosis, these patients are interested in having a specific molecule that precision medicine can target and specifically treat with few side effects. But what is important today is the so-called personalized medicine. So the patients with multiple sclerosis want to get access to the best possible care for them, not just as a former subtype of disease, but also as individuals. And I'm here referring to the perception of risk of therapy that is really varied in people with multiple sclerosis. And therefore, in the case of therapies with a higher initial risk due to the hematopoietic transplantation, or in the case of other more experimental therapies that have greater risks,
the unknowns, so things that we don't know and that we are not able to predict. So we need a communication channel in order to shed light on the individual. So I leave it there because actually I have an operational proposal, but I think that it will be for a second phase of this discussion. So I just wanted to add something pretty short. MESEMS worked. So that does not mean that you have to be grateful for what we did, but I will show because MESEMS worked, but we know perfectly that we tried to do something, so the working conditions were not perfect. So it took us 6 years to achieve a result. If we had one single production of mesenchymal cells, if we could have all the patients needed, so if we could have a trial done in a specific way, maybe. So maybe I never tried to ask Mario and Paola, we should let's have a new study. But by the way, we have a incredible preclinical data, and I don't want to think that preclinical data are good. So we had some problems, but so as you mentioned before, an extremely active subgroup of patients. So and we had in which there were relapse. I think that we have to see how we can transform this, translate this in a therapy. I really appreciate what you said before about the results that were pretty poor. But if you take this therapy and maybe you can make it up with something different, so with a little bit of makeup, we can improve the therapy. So thank you very much, because actually you have shed light on science, which is not the science which is a set of experiences, tests, discussions, trials, errors. We have a short time at our disposal. We were saying that the solutions are not simple, and maybe we should not speak about the solutions. So we should speak about the proposals or options. So hence, I have a challenge for you. In 1 minute, I would like to ask you to sum up a proposal, a feasible proposal. Gianluca Pedricini has the floor. So So our association, FISM, is an association of people for people, and this is our strength. This is our strength, and this allows us to play an important role. This allows us to talk to the national authorities in order to explain the problems of people affected with this disorder, this disease. We have to boost, to increase the participation in this kind of studies, in the multi-act studies, because if we increase the participation, the data will be objective, and those data may contribute to give an analysis and an assessment on the impact on us, and also an assessment related to the affordability and the sustainability of the advanced therapies. As a way of solution, we can get started with this roundtable, which is a multi-stakeholders roundtable. This could be the starting point in order to keep on working outside this conference room. We have to disseminate the results. We have to pull together all the actors. We have to create a sort of a big box in order to elaborate tangible proposals. Thank you. Angela Martino, yes, I will be really short and shorter than Gianvito Martino has spoken about common work, which reduce risks for everybody. This is true for the information side, because if we work together, we involve the patients, because the patient involvement is a scientific thing, is a medical thing, is a health-related issue, because today we have the strength, we have data at our disposal, data related to a community of people that learned to read the needs and to understand the needs. And also we have a room for maneuver in order to disseminate these results outside our community. Let's work together in order to improve our communication, in order to streamline our communication, and in order to allow people to make the right choice. So, and also, so I was really impressed by what you said about the costs. I think that when we speak about the costs for a person, for a disabled person, I always say you have to look also on the other side. So you have to think about the social cost of a person, of a disabled person. So you should disseminate this information, because actually if those advanced therapies are useful in order to avoid people with multiple sclerosis to become disabled, those therapies should be disseminated and developed. So I will be really short myself, and I will be. I want to stress the role of people in the research in all the phases, because sometimes we speak about some phases of the research. People, individual, is really important. And this is useful in order to provide with reliable data, and also to make a contribution to the different steps and the different phases of the research. Gianvito spoke about the contribution made by each and every individual in the research, and also the costs. We have been saying that we have to release resources with drugs, generic drugs, in order to resources that should be invested in drugs works for other people. So actually today we said that what we earn will be invested in innovation. So obviously, I don't think that in the light of what we have said before, we don't understand the role of a foundation such as ours. The role is not only, the role is an active role to make a contribution in order to carve out tangible solutions. So as far as we are concerned, there is another task, which is to mobilize international resources, international funding, because actually research is stem from an effort which is aimed at creating an international consortium between our associations in order to have money to fund the research. And so this is important. And beyond all the discussion about the rules, standards, so we need the standards. And what we do with less resources, but with the same effectiveness, may be supported by the system. As you have said before. Very last thing, since an awful lot of people are listening to us remotely, how can we get the funds from fundraising activities? I don't know, but I think that thanks to this roundtable, we have proven to be effective in investing the money. So we have to create new resources. Also, public funds are really useful. Are really important. Paolo Mauraro has mentioned an activity pulling together 8 billion euros and of our foundation and 8 billion euros of coming from the ministry. So we are able to use to make good use of the funds. Please support us. So I would like to dwell on what you have said before. So the proposal to create a system starting from the research and also the role played by foundations, associations in working on the strategic, international strategic agenda about the financing of research, because we need a good structure in order to support public research, which is important in order to develop the industry. So we have to create a system started from the research. So I think that all the stakeholders have to be pulled together, patients, regulatory bodies, researchers, companies, in order to carve out the pathways going from the research to the patient with real therapy. And we have to make a good use of the, so in this, we will have a common discussion about the way an experimentation should be done in order to have a drug, in order to convince a patient, to convince about the sustainability of a drug. Thank you. So I would like to dwell on what we have just said about the financing, the funding of a public research leading to a common research. Italy, with respect to other European countries, has a very low funding of research. Resilience Recovery Fund helps, but Italy has not been able to create a stable network of research activities, and Italy has not been able to attract huge investments. So we have to keep on working together with the institutions in order to boost the development of research, but we need important funds. I think the foundation does a terrific job, but I think that the foundation should not not be left alone. So thank you. Gianrico Martino. So I will be really short because actually we are talking about some problems that are the symptoms, so that these are not the causes of a system which has a huge cultural problem, because in Italy nobody gives a damn about the research, which is not good, because research should be an integral part of a natural cultural heritage. So I think that the system should be renewed since the schools, because actually we have to teach people how to interpret science. Rita Levi Montalcini was among the first person creating the scientific kindergarten, because she said that it was really simple to explain geometry, for example, to our kids. So we have to make people aware about the fact that the research is an integral part of our society. Research is a way that we can use in order to better understand the functioning of our nature. It's important to, and I fully agree with what my colleagues have stated. We have to work together in order to learn together, to face our criticalities. So this is the only way forward, working together. Antonio Celli. I will be really, really, really short. So working together, and I would like to stress one thing. I would like to ask you, how many of us would say, I don't want to cooperate, I don't want to create partnership? But the partnerships should be serious, and should be serious. I have been working a lot with patients with MS patients. So I have been working on the research in multiple sclerosis, and I've been working with the industry, and now I have a role in the field of the governance of public institutions, and each and every person, each and every actor has a specific needs, and needs that are crucial, but have to be pulled together. So this is something that we have to do in a clear-cut way, in order, because we have to avoid big statements leading to nothing. We need tangible results in order to impact on people's health and on people's life. Thank you. Paolo Morano. So I think that we have already stressed all the issues, and I have a proposal, a very simple proposal, tangible proposal. I work in the field of lymphohematopoietic transplant, but I think that the same technologies should be applied in other fields when we will have all the clinical indications. So how we can read in the Charter of Rights of MS patient, those patients in Italy have the right to have access to the lymphohematopoietic transplant, and the right to have access means that for the person, we have to double check if this transplant is good, but if a patient can receive this transplant, the transplant, he has the right to have access to it. So he or she has to receive all the information and also be treated in a center. So FISM should ensure that the network of centers that already exist, network of centers with a focal point, if you like, with a contact point, and the contact point should ensure a continuity. So, a contact person, young and senior, in order to ensure the continuity with the relationship with the patient. Thank you. Okay.
Allora, siamo arrivati
So we have reached the end of this round table. I would like to thank all the discussants. I would like to thank all the participants. And I would like to tell you, so we have been speaking about the science, real science. We have put on the table an awful lot of proposals. It's a pity because actually there is one of the key actor. So there is not. So the decision-making side is not represented in this table, but I think that we have to communicate all these needs that we have to take into account in order to create the partnerships, the networks. I think that we can end with this round table with a good hope, a big hope to keep on working for the present and the future. Thank you.
Ricordatevi alle 2 che ricominciamo le sessioni. Sì.
Com'è andata? È andata bene? Sei stata soddisfatta? Sono stati tutti bravi?
Allora, buon pomeriggio a tutti. Come sapete, come abbiamo detto,
Good afternoon. As we said 2 days ago, we want to underline our commitment to what we initially defined as related diseases, because Gianluca Pedicini said that we must say multiple sclerosis, optical neuromyelitis, and other diseases, related diseases. And therefore, we follow his instructions. Last year, as you may recall, we had a session with various speakers that took stock of the situation in the field of research on optical neuromyelitis. We had a newly established meeting of optical neuromyelitis, and now we have a lot of members. And on that occasion, there was a question and answer session that you can still find online, the possibility of taking stock of the solutions found so far. Last year, for example, we awarded the Litarevi Montalcini Award, and the winner was Matteo Gastaldi. So Matteo Gastaldi can come to make a report for rethinking the diagnosis of multiple sclerosis-related disorders, NOMSD, MOGAD, differential diagnosis, and so on and so forth. Thank you so much for the kind words of introduction. So I'll try to comply with the time allotted to me. Thanks to FISM and FISM for giving me the possibility of presenting a topic by which I set great store. And, you know, talking about these diseases provides food for thought. And I want to tell you some stories, some of which have been published, some others have been particularly successful, because I want to reflect on these type of diseases. Obviously, the starting point is multiple sclerosis. Obviously, I need not elaborating on what multiple sclerosis, pathogenesis, and antigenes are. But what has been particularly fascinating for me, and this is the reason why I'm interested also in a related area, in spite of the extensive amount of research, what has always been missed was one single antigen, probably because there is no one. But you may know that over the years, the effort made to identify the antigen for multiple sclerosis has been particularly frustrating. And indeed, of all the candidates proposed, no one was, in a sense, considered to be the antigen for multiple sclerosis. Today, I like to talk about autoantibodies in multiple sclerosis. And in my opinion, this is particularly important and can give us lessons, because, you know, it is interesting to stress, and Luca Battistini said yesterday, there are some indirect evidence and data regarding, for example, monoclonal antibodies. And you may know that in the multiple sclerosis, we have some deposition of antibodies. And there are also interesting evidence and data regarding the fact that if we tackle the problem, taking into account samples of liquor and comparing it with in vitro and animal models, we can see that there are some non-characterized antibodies. So certainly there are some interesting aspects that should be considered, but a notion that is so important in the stories that I want to show you is a concept that is now particularly important, namely the conformational targets in autoimmunity. So we have some direct responses, for example, sequential amino acids, but we also have direct responses of amino acids that are close to the tridimensional structure of proteins. So what has really changed this scientific field is the fact that now we are focusing our research on these issues, and we have almost 90% of this reactivity that we have studied. And therefore, we have adjusted to this situation, developing some tests that are some immune assays that have these conformational antigens. So I'd like to show you some pictures of the movies of David Lynch. The Straight Story is the story of a man crossing the United States to visit his brother. And it is a straight storytelling, I would say, that starts with a way to carry out research reminiscent of the traditional approach of the analysis of clinical phenotypes. For example, there is an involvement of bone marrow, optic nerve, and the most successful story was that of the neuromyelitis optica. And there is a debate. This is a sentence by Russell Rhee of 1930. There were people that thought that this was just a variant of multiple sclerosis, and others that thought that it was a completely different disorder. So there has been the identification of a marker of all the people that had these problems. That is this AQP4 antibody. And starting from the analysis of patients with a specific phenotype, we identified some reactivity that were found in specific areas, and this was identified as an activity against aquaporphyrin. Here you can see AQP4, where you can find these antibodies, and therefore you can easily understand the importance of such a specific marker. Today, we no longer talk about neuromyelitis optica, but of spectrum disorder. You know that there is obviously the involvement of the optical nerve. We know that there may also an involvement of the bone marrow, more than 2 segments. And we also know that we can have the involvement of other areas of the nervous system that reflect the areas in which the distribution of AQP4 is evident. We may have, for example, some lesions in the brain with some effects of narcolepsy. So you can consider that in this spectrum, we have many different symptoms. And this is not just a clinical marker, because this also tells us what the development is. If the disease is not treated, we will certainly have continuous relapses. But then we must move from clinics to pathogenesis, because we know that the direct link of antibody has an effect, a pathogenetic effect, because the first thing that we see, it is an internalization of AQP4 on the surface of the astrocytes, and therefore neurons and astrocytes have problems of lesions due to the glutamate, and therefore it is hard for them to remove this glutamate from the synapses. And we know that the link of the antibodies IgG1 fixes the complement, and destroys the astrocytes with other related events lead to a secondary demyelinization. And what we know now is that certainly the antibody is the main actor, main player of this disease. And you should think that this narrative tells us to know that now we have 3 drugs approved for this drug, having high efficacy. Drugs that have a significant impact on disability. Improved results have been achieved, and these happened in a period of time, let's say 15 years, in which the disease was defined, and then they also approved very effective drugs. So I think that it is a linear success story. So now in this scenario, we have an important marker that is the perfect biomarker, and therefore we could extract neuromyelitis optica from the multiple sclerosis area so as to consider that disease as a disease in itself. And I think that this is certainly important because we must push us to continue research. Then we have stories that are more ambiguous, so to say, in terms of research, in which the storytelling is not so linear. And we have the so-called anti-MOG antibody. This is a very known antigen that is linked to multiple sclerosis. This is an antigen that, from the percentage viewpoint, it accounts for a small percentage of proteins, but it is expressed on the surface, and therefore it is accessible to an immune attack. And you see, this protein is used to establish the experimental model of multiple sclerosis that is AIA. And so these anti-MOG antibody are investigated. And in 2003, indeed, we made this qualitatively forward so as to establish the presence of these antibodies in patients having multiple sclerosis or liable to develop multiple sclerosis. And, you know, the crucial aspect are the importance of the antibodies, and indeed the determinant of inaccurate or inconsistent results is that we had not carried out an appropriate standardization with appropriate tests to identify the anti-MOG antibodies. This is a study carried out together with Diego Franciotti, and we have reached consensus, and there was an attempt of standardization in which the various centers tried to use their tests to see whether they had the same positive results. And indeed, there was no specific overlapping or agreement. This data is particularly important because they've never been published. But then the story of the anti-MOG antibody changed. This was a narrative that was already known. So using conformational tests, those antibodies were identified not in multiple sclerosis, but in patients having other diseases. And once again, the aggregation power of this biomarker was evident, and now we are starting to consider these as a clinical spectrum that is separate from multiple sclerosis. And therefore, in this clinical phenotype, we can aggregate various aspects. We have a neuromyelitis optica, or the involvement of the bone marrow with different characteristics. And then we also have other expressions that may be more difficult to identify. There is, for example, the ADEN antibody, because more than 50% of children that have this form of encephalomyelitis are anti-MOG positive. We have also the problem of cortical encephalitis. So this depends on the alteration of the gray matter with episodes of epilepsy. We are considering this phenotype as an homogeneous phenotype, while we do not understand why an antibody can express in this cortical way. And it is curious to stress that this can also tell something about the story of this association. As Roberto said, it is true that every model has its own limits. And therefore, as already said yesterday by Luca, we know that when we compare it with the pathological samples of people with MOGAD, that this is the perfect model of another disease that is exactly MOGAD, and so many drugs have been approved on the basis of these results. And then we also have the aspect of the pathogenic effects. This is not a linear story as the AQP4. They can activate the complement. In the experimental data we have, less than 10% of patients have a significant complement activity. So this is not certainly the main pathogenic mechanism. On the basis of animal data, we know that the antibodies can activate through the PC gamma receptors the microglia, and this data has never been confirmed for humans. So we have weak data in this respect, and other experimental data stating that the antibody favors the penetration of T lymphocytes within the central nervous system. But certainly evidence of the pathogenic effects are still limited in scope. And there is another important aspect that must be stressed, because, you know, the linear storytelling is more widespread. We know that considering the various tests that we have, the best tests available, that we have patients with multiple sclerosis that also have these anti-MOG antibodies. We must consider all these biological effects, and we must better understand what happens in the pathological development of multiple sclerosis. So in this second area, certainly the virtuous work carried out by the anti-MOG antibodies enabled us to identify another aspect that should be better defined. So our marker is far from being a perfect marker. It is a useful marker, and there are some other aspects that must be further investigated. Then there is another story before approaching my conclusion. It is the story in which we must consider that sometimes our first impressions are completely wrong and can also be misleading. I think that you all remember this story. In 2014, another antibody was identified that was considered to be particularly promising in the field of multiple sclerosis. And it is a story similar to the one of the AQP4. And therefore, on the basis of sound data, we know that these antibodies are present in these patients with multiple sclerosis, 50% of these patients. And these antibodies seem to identify the effect of this AQP4. And therefore, in the method that we use to identify the antibodies, we may have completely different stories in which these antibodies are absent or were present, distributed in different areas.
This is a reminder of the fact that when there is this narrative. We have to take into account solid tests, standardized tests, because otherwise the message is not effective. So take-home messages, I think that they are pretty clear. First and foremost, we have to reconsider all the group of diseases of this kind and how some diseases have taken out a big box. There are some so with respect to the antibodies, we have to understand who are the other pathogenetic actors. And also we have to understand the role of antibodies and the interaction of antibodies with the cell immunity. So all these researches have to be based on the standardization of antibody tests. We have to so I think that our mind should be clear in a way that it should be wide open, because we may have this kind of scenarios, an antibody identify in a disease, and also we may have an antibody defining a disease, and we can find this antibody in other circumstances. And also a further scenario could be more complicated, such as the one of sclerosis, multiple sclerosis, because the antibody can be one of the reasons justifying the disease. This is something that we have to understand in the future. I do like the attitude by FISM, for example. Think about this spectrum of diseases in which you have at the heart multiple sclerosis and other diseases, and also there is a gray area that we have to better understand. Thank you, everybody, for your attention.
Bellissima presentazione.
Thank you for your presentation. So as far as the activation of, is there a difference between the diseases? It's a really interesting question, because actually we know something. If so, there is data for the KR4, those data. So the activation of the complement is a feature which is represented. So this is one of the main mechanism, and also there is other. There are cell models concerning. So we can see that the data are between 5-20%. So we do not understand if the mechanism is relevant. So if JJ1 antibody is able to activate a complement. We have to double check, because we do not know the relevance. So the complement exists also in MOGA.
Matteo, grazie, bellissima presentazione. Hai detto c'è il 5% di persone.
So you have said that there is 35% of people with a MOGAD positive MOGAD. Do we have this figure for the for healthy population? So if do we know if there are some protection mechanism? So very interesting question. In the healthy population, we know that in the optimization of the test, we can say that when we have a quantitative cutoff, because and the test MOGAD is based on a quantitative cutoff, so we can see healthy people below the threshold. So I think that in healthy population and also in not healthy population, we have So I was talking about high titer antibodies. So I think that this is the most interesting part. There is no studies now aimed at understanding if there are protective mechanism, and also in the laboratory, MOGAT is not so simple or clear. Thank you. I give the floor to the two coordinators of the round table, Roberto Bergamasti, that I see here, and Davide Cattaneo. Okay. This is the last session. Last but not least, because the agenda is really interesting. We are talking. The issue is brain health, rehabilitation, and public health. So we have some very interesting interventions. We kindly ask to limit your time of speech. So the first speaker is Gianpaolo Brichetto. Nobody knows him. Gianpaolo Brichetto will speak about promoting brain health in MS, an holistic approach. Well, I will deliver some introductory remarks with respect to what we will have afterwards. And so, first and foremost, some literature evidences about the rehabilitation approach in multiple sclerosis. Here, this is Cochrane Reviews, which has been published in 2019, and it is stressed the importance of rehabilitation in order to improve the situation. Physical rehabilitation, workout. So this is clear cut. This is pretty clear, namely because it has an impact on the reduction of deficit. We are talking about fatigue. So we have seen that fatigue is really important among the problems, the mobility-related problems created by MS. And also an improvement of functionality, namely as far as muscle strength is concerned, and mobility, and also the improvement of the quality of life. And beyond that, there is further evidence stemming from the Cochrane reviews related to the holistic approach, rehabilitation programs involving different health operators, and for outpatient or also hospitalized patients, there is a long-term improvement of the activity and participation of these patients. So with respect to the ECF, we can say that the effects of the rehabilitation it has an impact on the body, the activity, and the participation. So research in the rehabilitation field tries to explain the behavior-related effects of the holistic approach of rehabilitation, but also the research tries to show an improvement for people with MS. So there is a work which is aligned on the title of the presentation. This is a work by the Giovannoni Group in 2016. They stress the importance of a brain health approach for multiple sclerosis, and also it should be an early approach. And also there is a further aspect, further element which is related to the evolution of this disease, and also how rehabilitation can act on the evolution of the disease, and namely on the neurological reserve. And also there are some evidences related to the brain atrophy. So in this slide belongs with the Nobel Prize Rita Levi di Montalcini. So rehabilitation has a biological effect on the white and gray matter. This is something that we have demonstrated in animal models. This review has been published by Italian researchers and a Belgian researcher, and it has been published in 2023, and here they score the biological effect of rehabilitation on the central nervous system in a model, in an animal model of MS. So it's really interesting to see how the absorption is the same level of workout for a progressive multiple sclerosis. So there is this the biological effect on animals is the same that we can find in persons, in individuals. This has been published in 2018 by two researchers that received the prize Rita Levi Montalcini, and they stress the biological effect on the central nervous system, biological and structural, stemming from some fields of rehabilitation leading to the cognitive rehabilitation and to the motor rehabilitation, walking rehabilitation, physical rehabilitation. And also, there are some important options when it comes to the research that we have to do with in the field of rehabilitation. Here, there is another work which has been published by a Danish group, and this work analyzes the literature in the field of rehabilitation, and the premium is set on the target of population. So all the works that have been published has been based on a disease diagnosed 5 or 7 years old here before. So there is a window of opportunity that we can use, we can exploit on the early stage of the disease, at the beginning of the disease. We think so. I think that there is an awful lot of things to do in this context, and also there is another field in which we should explore the effects of rehabilitation is a progressive and advanced level of disease. So we can see that the studies are really few. Something has been done. I don't want to anticipate the results, but an international trial has been closed in recent times, and it's an international trial devoted to the progressive forms of the disease. And basically, in my short time of speech, I have tried to underscore the effects of the rehabilitation, physical exercise, and of the holistic approach when it comes to an MS patient. Rehabilitation is important not only from a physical point of view, but also from a well-being, from a physical point of view, but also for the well-being of the patient. So I would like to thank all my team, the team working in the field of the scientific research, all the institutions that have collaborated with us in our researches, and I would like to thank all the persons that have been involved in the research projects. Thank you. Thank you, Gianpaolo. So we have the opportunity to raise some questions because you have saved some minutes. I don't know if you have questions to make. So thank you for your presentation. Thank you for your presentation from a specific standpoint. We are reassured because actually there are some evidences about the results of rehabilitation. But on the other hand, if we had to have a notice of indications and contraindications of the rehabilitation, so there is an awful lot of things to know. So I would like to ask you, with respect to the three fields that you have mentioned, I would like to ask you if we have to boost our interest, our commitment in these fields. Actually, this issue is really important because on the one hand, we speak about medicine, rehabilitation, physical rehabilitation, customized rehabilitation on the one hand. On the other hand, we have to standardize our procedures in order to achieve tangible results. In this context and this framework, I think that there is an awful lot of things to do. First and foremost, we have to establish the right dosing in the treatment of diseases through rehabilitation, and also and also we should establish a rule, a standardization of the treatment based on rehabilitation. So it's a very complicated work that should be done. Thank you. Okay. So I think that we have the second presentation. Andrea Tacchino, same group, and he will speak about wearables and apps to monitor MS. Thank you, because actually my task is my report is about wearables and apps and how we use these devices in the research that we carry out in our laboratory. As we know, over the last years, the digital revolution changed our lifestyle and also our professional style. As it is said in The Economist, the wearables have the capacity to connect our us with our everyday activities. So they allow us to measure our activities through some new indicators. And thanks to some apps, we can interpret these indicators that can collect data in order to improve the effectiveness of our research, of our study. Everything is helpful in order to improve the diagnosis and the treatment, and obviously this is something crucial for clinical and research activity.
If we look at the projects funded, we can note that the foundation has really embraced this technological revolution. Technologies are particularly important, especially in the field of rehabilitation, and in particular, digital technologies are increasing their role. From the viewpoint of our research sector, when we use new technologies, we do so with a multi-layer approach. We can start from what we have in literature and the needs that we have to meet, also considering what we have available also in other labs with which we cooperate. And in some cases, we also developed them in-house. But our approach is based on a first step, the user test, that is the involvement of people with multiple sclerosis in the early phases, so as to have more updated versions and then test the feasibility study. Basically, this can provide indications on how this can be used in the future in daily activities. We can also have primary outcomes that can be tested in randomized and controlled trials, so that these devices can be used also in a clinical setting. Then we can also enter into the details of the cases in which we use this technology. As to apps, we developed this app, Cognitrack, that was developed 13, 14 years ago, thanks to a project funded by Professor Marco Bove, in which we developed an app for cognitive rehabilitation, because at that time we only had software that could not meet the specific needs. It was a sort of adaptive treatment. And on the other side, this was also an intensive treatment. Some results were achieved, and then we have updated versions of this software used in a special project by Professor Amato in pediatric patients. Another example is the app about CAM. We developed it in the framework of a project to assess the equivalence of outcomes in a standard format and in a digital format. Indeed, we used these portable devices, but there was no real evidence of the results. And then we realized that we can also see whether the responses may change. And then we have another app, DigiCog BES. Dr. Podda will certainly talk about this app. It was developed thanks to the collection of funds and was also awarded a prize in the Maker Faire. It is an app that is used for carrying out motor exercises, supported by the use of a sensor, and people at home can carry out this exercise following profiles which are monitored through the sensor they wear. What is the idea of this project? The idea is that of having the possibility of carrying out this type of exercises at home. So the basic idea is the so-called continuity of care. Another study that we are developing is a cognitive teledetection system. It is a commercial application whereby the person at home connects with the operator, and the idea is to assess the efficacy compared to a standard treatment in person. There is also another project, and we are working with Professor Casadio, a driving simulation that has been particularly appreciated, and we hope that next year we can show you the results. From the wearable devices viewpoint, we know that the most used ones are the inertial sensors that we use in a first study funded by RIMS. It is a European multicentric study on the dual task, so the cognitive motor interference when you carry out exercises requiring both a motor and a cognitive component, thus assessing what happens when you carry out the assessment. Similar sensors are used also in a study funded by Dr. Elisa Gervasoni to assess the effect of rehabilitation. And these type of sensors are lab sensors, while if we move to a real-life setting, we can use, as an example, the results of a project by Davide Cattaneo with the use of an activity tracker to check daily activities. And we see that through the daily activity, we can differentiate between people with multiple sclerosis with a low level of disability and healthy people, and people that have a specific form of disability in particular, because this shows that there are some differences that can be measured with some devices and apps. Now there is the project FT-CANE, a project funded by Professor Bove, in which the idea is having a sensor stick to check the movements of the person, and through some sound biofeeders, we can improve the walk of the person, and we can also enhance the efficacy of the rehabilitation treatment. Another example is exosuit. So this exosuit can be worn, and we are using them in the frame of collaboration with Professor Mazia in the Heidelberg University. And in a first pilot study, we have realized that using this microsuit for the upper extremities, we can reduce fatigue and improve endurance in the case of pick-and-place movements. We have just started the project on the use of exosuit for lower extremities to check the feasibility of the project during the treatment and also outdoor. Last but not least, the INHACT project by Professor Barresi. And in this case, the wearable used is a project to assess tremors in people with multiple sclerosis. The interesting aspect is that wearables on the one side and apps on the other provided data as well as model that enabled over time to implement this shift from a center-based model to a patient-based model. The fact that the person is at the core of this model and of the management of the disease, I think that can provide a continuous flow of data, objective data. This is exactly what we mentioned in this revision dating back to some years ago. On the other side, this work on the PROM initiative is that the wearables can improve the patient-reported outcome. These are two methodologies that must be further checked and can provide an overall vision of the person, and in this case, in particular, the person with multiple sclerosis. This union led to the participation in 3 big projects. The project Mobilize D, a world project involving many centers, including ours. We talk about unmet care needs, and we involve different groups of population, including people with multiple sclerosis, to achieve the adoption of wearables and apps for their management. Mainly focused on motor aspects is the project Alameda, a project with which we assess different functions, including the emotional state, the quality of life, sleep disorders. The person has many different devices used not only in lab but also at home that can allow overall monitoring. And results are about to be published. Last but not least, I'd like to mention the participation in the RAISE project, funded by the funds of the National Recovery and Resilience Plan. We are participating in 2 spokes of this project, one of urban technologies and the other one on smart devices and robotic technologies. Therefore, in this context, for example, it is better to talk about the spoke one. The idea is to apply sensors to the urban environment so that it can be checked, monitored, and there is the possibility of creating relations with citizens. Citizens are provided apps, sensors, and basically the idea is to create the so-called digital twin of citizens with multiple sclerosis, so that the services of the city can automatically match the specific needs of the individual. For example, a person that has to go out to go to a museum or to the doctor, they can suggest, for example, the best route to be followed in order to consider the level of disability, the environmental conditions. It is certainly an advantage for the future. Last but not least, I'd like to mention the results of these 2 surveys carried out with REAMS, one for multiple sclerosis patients and another one for the operators. And what emerges is that generally technologies are not so used, and there is a difference between the pre-pandemic phase and the post-pandemic phase. We have wearables and apps that are the most used, but probably there is room for improvement. We need to train the people, and we can do so through the associations and the healthcare professionals. So some take-home messages: apps and wearables are certainly consolidated tools that are needed to assess the motor functions and the progression of the disease and improve rehabilitation. Clearly, the integration of many wearables and apps gives us the possibility of get big data to develop models for multiple sclerosis. So we can open up new scenarios regarding a better personalization of the treatment and the possibility of simulating the intervention. So we can think, for example, that in the future we can simulate also the effects of some interventions. And then we must also consider barriers at the level of cost, usability of devices, and reluctance to change. We must also consider, together with this greater involvement of people with multiple sclerosis, the definition of better guidelines for clinical use. I wish to thank all the people that every day work with me. Thanks for your kind attention.
Thank you, Andrea, for this beautiful presentation. Beautiful and useful. There is a question, maybe. Thank you, Andrea. So this is not a question, this is a remark related to the future of neuro rehabilitation and the involvement of people, because actually we are talking about the DPI data about the preferences with respect to one device, such as this one, or the innovative intervention by persons. So I think that this is the best way to let all the stakeholders understand what does it mean, what it means to involve people in research, because we have to use this data in order to carry out rehabilitation protocols, protocols based on digital devices as well. So until we do this, it will be difficult to translate in health policy the use of these devices without speaking about digital devices. There is a huge difference between regions in Italy. So this is to tell you that the involvement of people is based on this, because in order to have access to treatment, we have to ask them the level of acceptability of the digital device and how these devices can be used. Because actually, if we impose those devices, I think that we won't have any results. If I may add something in this pathway, in this transition from the research. So the last sentence, the routine clinical use, there is another issue, because all the tools that are applied to the research, if we had to apply them to the clinical practice, they have to be certified as medical devices. And here there is another word, if you like, so that it's a different matter. So over the last years, for us as well, from the research point of view, it's really important to create a concept which is lacking. We can say, let's try to cater for a specific need. But needless to say that the use of the existing devices is crucial, but we have to certify the devices that you have developed in-house. And I think that the procedures should be streamlined in order to shorten the time to get the certification. I think that Davide knows this issue very well. So thank you. So the third presentation is Ludovico Pedulla. Ludovico Pedulla, who has been working a lot in a very important field, because actually he will. So this is the topic is rehabilitation and physical activity in MS. So I think that the tools and it's important to have the right tools in order to reach this result. Good morning, good evening, good afternoon. I'm sorry, everybody. Thank you for allowing me to speak about our projects. There are some keywords that you can see in this slide, and they go hand in hand with rehabilitation, physical activity, and sport as well. So rehabilitation, it's the use of multidisciplinary strategies in order to recover physical function and in order to improve the quality of life of people. And if we establish our foundation over the last 20 years has been supporting a lot the research in order to show the biological effect of rehabilitation. So there are some important studies carried out with the University of Genova, and we have established a joint lab. Here you have two works representing a milestone, because actually we have demonstrated the effect stemming from rehabilitation, and by using magnetic resonance techniques. So we have these works date back to 2014, and in 2016 we have tried to use the new technologies such as functional infrared spectrography in order to study the brain in movement. So it has been a long research, but I think that the message is pretty important, because actually our foundation has to ensure the quality of the results. We can say that over the last 5 or 7 years, we have been able to face criticalities related to the signals, the analysis of the signals. We can say that the use of a sensor, we can say that these devices are really useful in order to improve the rehabilitation and the recovery of balance and the use of crutches as well. And this has been shown in the clinical tests and the clinical trials, and also the improvement of brain activities in the prefrontal areas. And this leads to improve an improvement in using the crutches. So there is another way to study the effect of rehabilitation is to look for markers in the blood and in the saliva as well. This is a special project coordinated by Mrs. Prada, and here we studied these markers such as 6-interleukin before and after the rehabilitation, made of standard exercises or virtual reality exercise according to the group. Take into account the preliminary results tell us that at the baseline in the blood, in the saliva, there are differences in markers between healthy controls and between healthy people and people with MS. So that means that the technique of markers in saliva is very interesting. And before and after the rehabilitation, there is a change at the neurotrophic level. The results are not conclusive, but by the way, we can say that I can say that the increase of the sample is confirms these results, and we will have the final results shortly. So let's have a look at the activity on the daily basis. So the use of wearables, So I have a short video. So this project has been financed by the RRF, and here there is technological. So here we studied cognitive and movement reaction while preparing a coffee, for example. There are so we have established metrics in the movement, and also by using QR code in order to extrapolate thanks to other wearables, to extrapolate the cognitive reaction. And you can see also the red circle, it's a gazer, so it indicates the pupilla as a matrix of the effort. I've tried to map this, to do a mapping of this project, because actually on the vertical axis you have the target of our patients, mild disability and severe disability, while on the horizontal axis I put the framework in which the treatment has been carried out. On the left, laboratory; on the right, in a daily life. So here you have the studies that we have illustrated. So there is an heterogeneous distribution. We try to take into account all the areas. As far as the rehabilitation is concerned, we are mostly in the laboratory. So the physical exercise which is used during the rehabilitation, it's a structured physical exercise in order to improve one different aspects. So here, so these words stem from a conference of young people. We have asked the participants to create a link between rehabilitation and the word. So first and foremost, physical fatigue. So this is a study in which we studied good interactions between physical activity and fatigue. The tool is TMS, which allows us to study the inhibition of the central nervous system, and in this case we the inhibitor so you can see that if we stimulate this part indicated in red, so we may have a silence which is assessed at the peripheral level, which is so this is a silent period, as we call it, and this shows fatigue. So we have studied this data in healthy people. You can see that the silent period increases after an effort, a physical effort, because so there fatigue, there is an increasing of fatigue. So the next steps are based on the assessment of the alterations of this parameter, and needless to say that we will take into account the use of protocols of physical exercise, physical activity, in order to have an impact on these aspects. Between among the protocols that will be tested, we have training, interval training, with a project carried out with the University of Genoa and Joint Lab, and the aim is to take a protocol which is used for sport people, and that will be adjusted. We will use the device that you can see in this picture. These protocols are pretty demanding, but by the way, past studies tell us, there is this study of Giampaolo in which people with progressive MS and disability and a high level of disability received an intensive program through this device without any adverse effects. So this is feasible. The literature supports us, because actually there is no relapses in doing created by the physical exercise. And so Andrea already talked about this study, exercise and physical exercise, physical and activity and physical exercise. So there are studies, and I would like to stress the right-hand part of this slide, in which you have the effect of the fatigue induced by activities. So you can see that the preliminary results for people with multiple sclerosis. There is an increase of oscillations after training with respect to healthy people below. But after a period, a rest period, it is possible to recover the conditions existing at the baseline. So training and recovering a rest period are of utmost importance in order to establish effective rehabilitation protocols. So once again, so that there are projects indicated in this slide. We can see that exercises and rehabilitation always concern a laboratory setting. So if when it comes to daily life, we speak about the physical activity, which is all the movements of the body lasting more than 30 minutes. And so, here we have also work and other activities. So you have a cloud of words. There are words such as well-being, efforts, and I think that those words should be kept in mind together with the importance of this issue for people concerned. So, and also we have to consider that the guidelines of the WHO tell us that for healthy people and people with chronic diseases, the quantity of physical activity is the same. There is no changes. So we have to take a different approach. I mean, physical activity is not a therapy that we can use in specific conditions. But physical activity is a common thread, and it should be adjusted to the needs of each and every person. There are some guidelines for physical activity for MS. So we have carried out a survey in order to see if people with MS comply with the guidelines and which are the activities. So we compare the situation before and after the COVID-19. So we have shed light on the activities that are more easily that are easier to do for MS patient, and also we have established some recommendations for during the pandemic, during the COVID-19, and also after the COVID-19, in order to keep a continuity of the treatment. So as I said before, this is the daily life, and then we have the sport life. Over the last years, we try to promote the physical activity adjusted to disability and to MS as well. And there is a cloud of words stressing inclusion and empowerment. Empowerment gives us an idea about the role the importance of the involvement of people while deciding a physical activity protocol. This is a study that we carried out with the University of Pavia, and it is really important to receive an advice by the general practitioner when it comes to sport. People receiving this advice are more tended to do sport, regardless of the disease, the age, and so on and so forth. Here are some initiatives that that we have been carrying out in order to promote the physical activity. There is a wind of change training, a sort of training that took place in the Isola d'Elba in 2018. It improved the quality of life of people involved in this project. And then there another event organized in Genoa in 2019 with so per people, you can see people with MS being involved in sport activities at the very high level. So we try to carry out initiatives involving people with different level of disability in those activities that you can see here. To conclude the projects that we saw, so those projects fill the spaces in the diagram. So this is the first take-home message. As a foundation, maybe our task is not to deepen. So we have to provide an answer to people having with different needs. So the importance of transnational research, which has been stressed in order to be more effective, in order to involve all the right stakeholders. I would like to thank all the staff of FISM that allowed us to develop the studies that I have mentioned before.
Grazie Ludovico per l'escursus completissimo della riabilitazione.
So it is extremely important for us to have this conclusion on the fact that physical exercise can be carried out in a more joyful and attractive way for patients. And indeed, I must say that obviously you mentioned this idea of amusement, of joyful activity. Are there questions? Oh, you've been very clear.
Quindi finora si è parlato di attività soprattutto motoria, riabilitazione motoria.
Now I'd like to give the floor to the next speaker, Jessica Podda. She will talk about the role of cognitive rehabilitation in multiple sclerosis brain health. Thanks for these kind words of introduction. Good afternoon. It is a great pleasure for me to take the floor on the occasion of the World Day on Multiple Sclerosis, and this is the reason why I want to thank Dr. Paola Zaratin for giving me this opportunity. In these 15 minutes, I'd like to give you an idea of the research project supported by our foundation for cognitive rehabilitation. And I'd like to introduce this issue with a sentence by Rita Levi-Montalcini. She had already mentioned the fundamental role of the mind having priority over the body. And so these two pictures, probably many of you may know, belong to this portraits campaign that showed the invisible symptoms that have a negative impact on the quality of life of patients with multiple sclerosis. And these visible symptoms are shown in this slide. This was the picture used also in the first meeting in which I participated, because, you know, the Asperger shows the complexity of cognitive experience. And in all these aspects, I think that this is the priority of international research. We must identify effective strategies to assess these cognitive difficulties. We know that there are some cognitive faculties that are mainly altered, and indeed the great variability of the situations of these patients show that there is a typical dichotomic approach that can enable us to differentiate between those who have cognitive difficulties or problems and those who have not, provides only a partial idea of this complexity. And this is the reason why over the last few years we had evidence of a new approach, a cognitive taxonomy. The cognitive phenotypes are patients that have a similar cognitive functioning. Based on the study of 2018, many researchers focused on reducing this cognitive gap, and the same applies to the foundation. In a study of 2021, we identify cognitive phenotypes based on the patient-reported outcomes, thus combining the cognitive and the emotional spheres. And you may know that, for example, mood disorders affect a very high level of patients. And so we want to identify a continuum, namely a phenotype with limited and isolated cognitive difficulties. And this is characterized by young people. And then we have a cognitive phenotype in which these cognitive abilities are manifold, faced with an increase in disability and an increased age. Over the last few years, we have also identified some neuroanatomic strata that can explain cognitive phenotypes. We can think, for example, to the significant contribution of clinical phenotypes in multiple sclerosis. On the one side, we can better know the symptoms of the people with multiple sclerosis and also identify rehabilitation and pharmacological treatments tailored to the various forms of multiple sclerosis. Clearly, in order to identify cognitive phenotypes, we need a lot of data, and obviously technology can help us to collect this data. And I'd like to tell you something about DG-COG-MS, the development of an app developed by our foundation. And this is used to manage cognitive difficulties. But you know, we have evidence supporting the usability of these new technologies, as was previously mentioned by Paola, are really limited. And indeed, we have first-hand experience of how sometimes we are not so inclined to use technology when it is too complex and unable to meet our specific requirements. So we started with data collected with the parameter, the issues raised by the patients with multiple sclerosis to have access to care during the COVID pandemic, for example. And there was a positive note, the potential of telemedicine to reduce the physical distance between doctor and the persons. And therefore, DG-COG map can be downloaded. And as mentioned by Andrea, DG-COG MES was carried out thanks to the contribution of streamers that adhered to this campaign, Stream for AIMS. And every year we give an award to the various technological applications that improve the problems of patients and their caregivers. This app is characterized by 4 digital tests that measure the impaired cognitive abilities. They are modeled on traditional tests that are used to assess cognitive symptoms. Recently, our validation study was accepted for publication, and this is the most important result because there is a positive correlation between the digital test of the app and the paper tests, the traditional tests. And there is also a good level of usability as shown in these scores. And I would also like to stress that the scores of usability are affected by the symptoms of anxiety and depression. And therefore, by conclusion, digital mass could be used also in clinical practice, although there are some difficulties to be overcome, as an additional tool in the toolkit, so to see, of neuropsychologists. Furthermore, all researchers that want to develop digital solutions for people with disabilities should consider the psychological and emotional aspects that sometimes are neglected. The assessment enable us to identify adequate treatments and also monitor the effects of our treatment over the last 20 years. As Gianpaolo said, we collected evidence on the efficacy of cognitive rehabilitation for MS patients. Indeed, we have little evidence on the progressive forms of multiple sclerosis. So in order to try and bridge this knowledge gap, the study COGEX, gathered 11 centers in the world to see whether cognitive rehabilitation and aerobic exercise could be effective for managing cognitive difficulties. The participants, over 300, carried out an accurate analysis to assess and measure some cognitive and motor aspects, for example, fatigue, anxiety, cognitive disorders, depression. And based on this assessment, patients could follow 3 different types of treatments, integrated treatments. So the cognitive aspect could be active through REGM, that is a computerized software that is used for cognitive rehabilitation, or passive cognitive rehabilitation. So techniques and methods to better use PCS to the motor rehabilitation. We had a cycle ergometer that improved the performance of the upper and lower extremities in a safe way. And then there was also a passive rehabilitation with the stretching and balance exercises. So what is the result of this assessment? We found no additional effect of the active rehabilitation group. So our participants improved in terms of speed of processing of information, regardless of the group they were assigned. And this is positive because COGEX is the first international initiative focused on the progressive forms of multiple sclerosis with moderate disability, and where the primary outcomes were the cognitive disorders. And this was the best and unique way to participate in a study in which commitment and involvement were high. And the unexpected outcome for this passive rehabilitation suggests that if people remain active from the physical and mental viewpoint, they can also improve their cognitive performance. Another group of devices that seem to be particularly promising for cognitive and motor rehabilitation are the EXER games, these active video games that enable to improve with one single device both the motor and the cognitive aspects. Evidence shows that there is an efficacy that is comparable to traditional therapy. The motivation is certainly higher, as well as compliance. But there is a growing need to develop EXER games that are tailored to clinical situations and interests of the people. We can also mention this study by Luca Prosperini extremus to show whether the combined effect of exergames on the cognitive and motor spheres could improve the cognitive abilities compared to a typical software Cognitrack that was mentioned by Andrea. Let us show some results. The thought of having followed a treatment we can improve the processing of information. Another study along these lines is ENACT by Giacinto Barresi. The goal of this study is to identify and develop ludic or playful solutions with virtual or immersive reality that can improve the dual task. Here you have some examples of the exergames that have been developed within this study. You see, in this video, participants should play with this tangram play. I do not know whether you know this game. You need to assemble all these parts in order to achieve that figure. Game ability is another important system we have. We went to high schools to talk about these issues, and boys and girls had the opportunity of knowing who is behind the research and those who develop video games, and their task at the end of this cycle of lessons was to develop some exergames to improve the cognitive and motor abilities on the basis of some information they had regarding gamification and multiple sclerosis. You know, there is some skepticism linked to the usefulness of exergames in the field of rehabilitation, but we must not forget that play game is the first activity that we use in order to know ourselves and explore the surrounding world, and it is inevitably an activity that is forgotten sometimes. But it is a great window of opportunity to study, for example, the theory of self-determination. If I'm more competent, autonomous, and independent, I will certainly continue to carry out this activity that gives me well-being. So this is the great success of video games. By way of conclusion, I could say that the progress, scientific progress in particular, that is carried out by the foundations enabled us to foster access to continuous and high-quality care. A recent report of the World Health Organization reports that 50% of world population has no access to essential care services. So we must work hard all together, people, researchers, and institutions, to remove these inequalities and have a situation in which nobody is left behind. I'd like to thank once again Maria Alberto Battaglia, Gianpaolo Brichetto, Paola Zaratin, and all the colleagues and the people with multiple sclerosis that we use for our studies. Thank you. Thank you so much for your kind attention. Thanks. We can thank Jessica. We are late, so I do not know whether you have questions. I'm so glad to introduce another researcher, Michela Ponzio. She will talk about the employment in multiple sclerosis and the role of vocational rehabilitation. Don't worry. You can make your presentation. I wish to thank Paola Zaratin and Professor Battaglia for the opportunity given to me to make this presentation on this topic. This topic is a priority in the field of research and public health care. it is one of the two areas in which in the foundation we carry out intramural research. I'd like to show you what are the other areas in the field of public research that we perform, the analysis of needs, and whatever regards the quality of life of people with multiple sclerosis and their caregivers. These are only some of the works that we have produced. Another interesting theme is the theme of epidemiology of multiple sclerosis, and particularly the interest of defining multiple sclerosis on the Italian territory. So we have different collaborations with regions, local authorities, using, for example, the flow of current data in order to use some algorithms in order to map this phenomenon on the various territory. And then I also included the logo of this project of the Italian Register for Multiple Sclerosis, because there is also one of the two operational structures of this project, namely the structure for coordination and administration, working in close coordination with the other technical operational structure located in Milan in the Institute Mario Negri. And here you can see some of the works that we carried out with this Institute Mario Negri, some of which have already been presented in the conference. And then there is also another important aspect, that is the cost of the disease. And indeed, I've mentioned some works. For example, in Italy, we've been among the first to define the cost for rehabilitation and the amount of money paid by the patient and the amount of money paid by the National Health Service. We were part of a wider project that investigated the cost of comorbidities in multiple sclerosis. And now I focus on the topic of this presentation, that is employment and multiple sclerosis. This is important because obviously this is a disease that affects very young people, sometimes that just enter into the labor market. So considering that for everybody work is a right, all the more so for people with disability, as stated by the UN Convention, stating that all people also with disabilities have the right to choose the job they want and work in an environment ensuring equal access and equal conditions. So this is essential for social inclusion and for independence, because obviously job strengthened relations fosters social recognition. And we must also recall that the job is linked to the person, the family, and the society as a whole. We know that the employment rate in Europe ranges between 30 and 65%. The employment rate is reduced if there is a high level of disability. We must not forget that this is a significant cost also for society. The lack of jobs is a big problem, because 45% of the total costs of diseases are due to the loss of productivity. And therefore, for the association, this has always been a significant topic in terms of rights of people, in terms of advocacy, but also in terms of research. I'd like to recall a collaboration we had some years ago with the Carlo Besta Institute in Milan, this instrument MSQ Jobs. It is a questionnaire that can assess employment difficulties, investigating into difficulties linked to the mental and physical situation and the employment contexts. And now I'd like to focus on these two projects funded by INEI. The first project is the Prisma project, and the second one is Real SM. Both projects are coordinated by IRCS, Policlinical Hospital San Martino in Genoa, and there is the participation of the Genoa University with the occupational medicine. And you know, this nucleus has been consolidated also in the future projects with the Foundation Santa Lucia here in Rome. Few words are now appropriate. This is a project that has already been completed. These two projects are closely related, and the projects obtained through the previous project were the trigger for the future project, so that the funder could trust us and continue to invest money. The Prisma project analyzes the employment reality of people with multiple sclerosis so as to define models and improve inclusion on the labor market. An analysis was carried out on the situation of employment for multiple sclerosis patients. A survey was carried out on patients to try and identify the factors that may affect the job retention. An attempt was also carried out taking into account methodological, anthropological, and demographic data. And we tried to define the specific characteristics so that a good practice could be spread also to other situations. 4 systematic revisions, and here you have the results. This is the theme analyzed in international literature. This has enabled us to assess and highlight that there are some occupational outcomes that are not considered. For example, the issue of stigma, communication of diagnosis, difficulties in job barriers. All these issues are not really well investigated, all the more so that we investigated into these issues in the 2 other revisions that we carried out. And therefore, we can say that most of the subjects included in these studies, 80% in particular, reported this feeling of stigma. The barriers are mainly related to the type of job and the working environment. There are some important results that have been collected and that have been at the basis of the implementation of the future project. Obviously, we work with people with multiple sclerosis, and also thanks to this publication, we could identify the factors associated with the individual difficulties related to the mental, physical, or job organization areas. When we were authorized to publicize the results of this work, we had unexpected results, and there is so much to be done in terms of training on the rights of the people. We carried out a monothematic work on the issue of disability and work, and a training platform in which we organized initiatives with the participation of more than 100 people, 1,000 people, I would say. And then I moved to the second project that is still underway, Project Real, multiple sclerosis, and it wants to identify the possibility of vocational rehabilitation to improve job retention. The longer-term objective is to identify some protocols and pathways to provide care to these workers with disability. Currently, The state of the art is tackled with a survey with all the Italian rehabilitation centers so as to understand how these patients are treated in rehabilitation centers. And thanks to the fact that in the project we have involved Brichetto, we could involve also important representatives from an international network of multiple sclerosis. And the results will be also provided to international centers. There will be also another survey on the people affected by multiple sclerosis to assess rehabilitation needs. And the core of the project, as an experimental part, we want to identify vocational rehabilitation initiatives based on implementation science. When we have to implement a new intervention, we are ready that we can find some problems. And therefore, this is the important result that we must take into account. All this in order to define protocols and pathways. Part of this training is addressed to all the players and people that are involved in this project. I would like to give a definition to vocational rehabilitation that is hard to be translated into Italian. We mean intervention to people that can work, that want to enter or re-enter the labor market, that find it difficulties due to accident, impairment. By definition, this includes a group of people that, due to the very nature of this intervention, this study will certainly have a part of feasibility, and this is the flow that we've tried to summarize. We can start from an initial assessment that will be tailored and personalized. So we try to identify 5 job difficulties according to a scale that is used in occupational therapy. And then we have also included the presence of a social worker, because we want to place this pathway within the project of life. We have just enacted our Decree 63, talking about this project of life, and employment is essential in this respect. And therefore, we need to identify the value of work for each of these persons in their project of life. Then we will organize meetings to carry out a case-by-case analysis. We have identified, thanks to literature and many inputs from the previous project, 3 main areas: the rehabilitation area for managing employment performances and the psychological aspects that play a significant role in this respect, a reasonable accommodation area, so that there may be the changes in the environment, and an education and training component for people with multiple sclerosis and their employers. Horizontally, we have included this idea of manager, of a facilitator that can coordinate this difficult process and implement it. Clearly, there will also be aspects that are not really related to the figures that we have identified in this process. For example, the employer, the occupational doctor, and so on and so forth. Then we will have a pre- and post-intervention assessment to see whether the results have been fruitful or not. We are using this protocol in many different contexts. For example, we participated in the conference of employment therapists, occupational doctors. This project has been welcome. this poster was presented in the Congress of Occupational Therapists, and this won an award as best poster. I only want to say that the fact of having within the project the Italian Association for Multiple Sclerosis may be a driver, and we hope that the results that we achieve will be really disseminated to the institutions. I wish to thank all the members of this research sector of the public health entities with which I cooperate on a daily basis. Without them, all these results would have not been possible. Thank you. Thank you so much for your kind attention. Thank you, Michela. Thank you for using this methodological rigor in tackling this issue that is certainly particularly significant for the people with multiple sclerosis. I wonder whether Professor Battaglia would like to add something. Obviously, we can now move to the conclusions. Few words just to conclude this 3 days conference, very intense conference, rich in good science and possibilities for cooperating with all of you. And I believe that these events are really particularly important to promote this networking. In the Scientific Congress of the Italian Associations of the Foundation, we spoke of brain health and the need of rethinking the diagnosis of multiple sclerosis and related disorders. And I'd like to conclude by saying that when we talk about brain health, clearly, you realize what we have heard in the last session in particular is of fundamental importance for the promotion of brain health, to which we all aspire. So I wish to thank the colleagues from the rehabilitation sectors and from the public sector for the significant contribution they provided. And this must be better disseminated into clinical practice. Considering the first day of our conference, I wish to thank all the speakers that took the floor. In the framework of a global agenda of research, our ambition is to promote brain health and rethink the diagnosis of multiple sclerosis. We stated that the global agenda is committed to promote research in priority areas. We have seen that we are doing this through national initiatives, European and international initiatives. I wish to thank also the international speakers that intervened. Many of them remained for the 3 days, and they wanted to support the impact that the research promoted by the Italian Association for Multiple Sclerosis has and will have at national, European, and international levels. This is particularly significant in these days. We are about to vote for the European Parliament, and for us it is extremely important. And our research must be present, and I think that it will be there, it will be present for 3 main reasons, and this is my take-home message. First and foremost, we need to focus on the quality of life, thanks to Giancarlo Brichetto, the ability to be ever more present in the definition of European digital platforms, and the importance that this has on the decision-making process, on the treatment and quality of life of people. But I wish to conclude with the aspect by which I set great store. This conference has shown in different forms and in different aspects that we need to consider this new discipline, the discipline with and for the patient. This must be turned into science, and in so doing we can meet all the challenges with which we are confronted, and we can also move towards this brain health that is so sought after, and hence rethinking the diagnosis of multiple sclerosis. By way of conclusion, I thank once again all the speakers, all those who have helped to organize this conference. I would also like to thank Marina, Rachele, that have been fundamental for the logistical organization of this event. And clearly, last but not least, I wish to thank the people with multiple sclerosis. Thank you. Thank you so much.